Morning, everyone. Good to see you. I'm Sangeeta Bhatia. I'm a liver bioengineer at MIT. And on behalf of my co-organizer, Wolfram Gössling, and our partners at the American Liver Foundation, it's my pleasure to welcome you to the 34th annual Irwin Arias Symposium. This meeting has a remarkable history and legacy for those of you who are new to the crowd. It began over 3 decades ago when the American Liver Foundation wanted to honor Win for his contributions. And I think the idea at the time was to have a gala, big party, and Win, in his characteristic way, said, let's instead have a symposium. Let's bring people together from the science side, from the clinical side, and really bridge a conversation about how basic fundamentals can apply to clinical medicine and vice versa. And thus the symposium was born, and we've been having it every year ever since. The idea is to have a home for rigorous science, to expose trainees, I see lots of them here, to what we think are the beauties and the intricacies of the liver, and to spark new ideas and collaborations. Over the years, this has become a reality. This meeting has hosted Nobel laureates, heads of federal agencies and clinical departments, as well as early career faculty and trainees that are still on their way. And there have been lots of aha moments. One of my first collaborations in hepatitis C started at this very meeting, so we all have lots of fond memories. Wolfram and I both attended the meeting as trainees, and so we were honored in 2017 when Win passed the baton, literally on this stage. We did a baton passing to organize the symposium. And together with Heather Fleming, Heather, you're in the back, raise your hand, who really does everything. We have built upon Win's vision to bridge scientific and clinical disciplines in a few new ways. We've invited researchers from industry. We moved the meeting here to Cambridge to tap into this vibrant ecosystem. And then we put a renewed emphasis on trainees and their exposure and visibility. We've been intentional about building a diverse community, scientifically, geographically, demographically, because we know that that brings the best ideas forward. And we have today actually over 500 registrants, including those online, which we think is the biggest number yet. So the meeting has grown every year. We're really excited about that. I know there's a bunch of folks online, so we welcome you, and please make sure to submit your questions to participate in the discussion. In terms of what to expect today, we have a series of full-length talks interspersed with 6 trainee microtalks. And the microtalks are really snapshots of more fulsome poster presentations. You can access the full program using the QR code, which is posted on the signs outside the auditorium. And there is an opportunity to vote for a fan favorite poster over lunch. So please vote. As a special treat, later this morning, Win himself will offer a brief reflection on bile acids, which, as you all know, is an area where he made truly seminal contributions on the role of ABC transporters and inheritable jaundice, amongst other things. I've known Win for over 20 years. And I was just saying earlier this morning, I've never had a conversation with him where I haven't learned something. So I'm really excited for you all to share that with you. Finally, before we get started, some thank yous to the Broad Institute for hosting us in this space, our AV team, our exhibitors, Abcam and Stem Cell, and our generous sponsors, Alnylam, Ultragenics, and Ochre. Thank you also to Wolfram and Heather and the entire ALF team for their partnership and support, and to all of you who've taken the time to be with us. As my last remark, I just want to point you all to a poem by Pablo Neruda. If you haven't read it, it's called The Ode to the Liver. It's worth Googling. He's a Nobel Prize winning poet. And he called the liver his modest, organized friend and underground worker that no one sees or celebrates until it wastes away. And for many years, this has been true, but I would argue that the appreciation of the liver is finally coming of age, and we've turned the corner. So with that, let's enjoy the day. Thank you for joining.
Good morning, everyone. My name is Prashant Nagesh, and I'm co-chairing this morning's session with Dr. Manjari Trivedi. It's an honor to introduce Dr. Jennifer Lippincott-Schwartz. She's a senior group leader at HHMI and a global pioneer in live cell imaging. Her groundbreaking work using a green fluorescent protein, GFP, has revolutionized how we understand organelle dynamics and protein trafficking in real time. Her discoveries have reshaped modern cell biology and equipped the entire field with tools which most of us rely on today. Please join me in welcoming Dr. Lippincott-Schwartz.
Thank you so much for letting me present online. Very sorry that I wasn't able to come up to Boston today, but I was sort of feeling quite under the weather yesterday. Yes, I'm going to be talking, sort of bringing some cell biology to the sort of the field of liver biology, which, as you heard, has been, well, is not only the topic of this meeting today, but is really a fundamental system that's operating within many multicellular systems. The liver is a system that, in fact, involves a lot of innervation. In fact, the liver innervation is surprisingly extensive. And this is why the topic that I'm going to be talking about is, I think, possibly relevant to this audience today. Basically, what I'm going to be talking about is how the individual nerves are organized intracellularly in order to be able to impact long distance processes. So this is a beautiful little image of the liver, and I've just drawn in the vagus nerve, which is coming in to the liver and innervating it. So sympathetic, parasympathetic, and sensory neurons, fibers, are moving into this liver and basically integrating themselves to regulate hepatic blood flow, lipid metabolism, bile, and glucose handling, basically relaying liver status to the brain. Now, what I want to do today is to really describe how at the subcellular level, neurons are capable of playing this important role, not only in organs like the liver, but also the brain and other tissues. So just to refresh you in terms of the organization of the neuron, there's an input region called the dendrite, and there's a conductal region called the axon, which basically leads to an output. And it's this axon that is integrating itself into the liver. The input region is reaching out of the liver into vagal nerves, et cetera, to allow communication with the brain. Now, one of the things that's very important to appreciate with this unique cell, the neuron, is that many of its processes require calcium. And I've listed to the right bottom some of these processes, which include excitability, neurotransmitter release, synaptic plasticity, and gene transcription. All of these things are actually regulated by calcium. And that's what we're really interested in trying to understand. Basically, how can neurons integrate these calcium-dependent signals to enable long-distance communication for achieving coherent and coordinated responses? So basically, in the case of the liver, as well as in the brain and other organs, it's calcium signals that are being received from the input from a dendrite that's going to be communicated to the cell body that leads to an output into the axon. So how is this happening? So in order to sort of help you understand this in the context potentially of liver, but in my case, we're going to be focusing here in the brain because this is where the description of neuronal connectivity has been most powerfully investigated. Basically, what you're looking at here is a EM reconstruction of two neurons that are communicating with each other. I've highlighted dendrites and axons so that you can see But if we zoom into this little synapse that you can see off to the left, you can see this is where a lot of communication, and each one of those individual spines, which are small little structures sticking off the dendrites, communicates with different axonal synapses. And this is just one of those. But basically, if you zoom in on what's happening at that synapse, it's basically a very simple thing. The axon is releasing vesicles that have neurotransmitters. Those neurotransmitters are bind to receptors on the dendritic spines. Those receptors lead to opening of calcium channels, but also trigger a depolarization along the spine and down the dendrite to reach the cell body. Now, the question is that calcium that's released into that dendrite is triggering intracellular calcium signaling messengers. And so somehow that information needs to travel tens of microns to reach the cell body, to set off a communication pathway into the axon. This synapse to nucleus signaling is not clear. How is this happening? You have a depolarization, that's basically the speed of sound, moving from the synapse all the way into the cell body. But how does the calcium signaling coordinate these tremendous distances? And so one scenario that we've speculated on was maybe there's a structural element in the cell itself that can locally amplify and relay calcium signaling messengers over long distances. Something like a cell phone tower, if you will. So the idea would be calcium comes in response to a synaptic input. That calcium is then relayed through some sort of structural element all the way down the dendrite to the cell body. If so, what might be that structural element? Well, my lab has spent a lot of time studying this beautiful structure called the endoplasmic reticulum. It's the largest organelle within the cell. It expands from the nuclear envelope out to the periphery of the cell, and it turns out to be a very important calcium storage compartment within the cell. Indeed, the ER is operating as a binary system with the plasma membrane to regulate many calcium-based processes. So basically, the cell wants to keep calcium very low in the cytoplasm to the tune of about 100 nanometers, nanomolar, whereas in the ER, you have 1,000-fold higher levels of calcium. Outside the cell, it's 20,000-fold higher concentration of calcium. So the way that the cell keeps this low concentration of calcium in the cytoplasm is through calcium pumps and channels that are found on the plasma membrane or the ER that can suck up that calcium or transiently release the calcium into the cytoplasm to alert the cell that there's something important that's happening, like a synaptic impulse. So one of the things that we know is that the ER extends throughout the neuron, which is comprised, as I mentioned, of the dendrite, which is going to be a huge focus of the next 10 minutes, the cell body, which we call soma, and the axon, which is going to propagate an action potential that is triggered in response to synaptic inputs that travel through the dendrite. So could the ER and the plasma membrane be collaborating to regulate this long-distance calcium signaling in the neurons, which is so critical for allowing the neuron to convey information very long distances? In the case of the liver, from the brain all the way through ganglia into the liver system and back out. So in order to investigate this, an incredibly talented postdoc in the lab by the name of Lorena Benedetti basically started looking at the endoplasmic reticulum in dendrites in detail. And what she found, as you can see here, is that, yes, the ER is extending throughout the dendritic system. You can see it in the proximal dendrite, the medial dendrite, as well as the distal dendrite. And importantly, when she looked at the organization of the ER in all of these different parts of the dendrite, she saw a periodic organization. And when I mean periodic, what I mean is that there are regularly arrayed contacts of the ER with the plasma membrane at 1 micron intervals throughout the entire dendritic system. And this is just showing graphically in the posterior, medial, and distal dendrite. If you look at these contact site intervals, they are constant. And this is to the far right, you're seeing a focused ion beam scanning EM in a mouse hippocampal dendrite, where we're mapping out the sites where the ER is periodically touching the plasma membrane. This is illustrated even more dramatically in this FibSim segmentation of a neurite in a fly, where we're just in green, we've mapped the structure of the ER in a portion of this neurite in the brain, and you can see a beautiful ladder-like organization of the endoplasmic reticulum, where it's splaying out all along the dendrite, where the edges of the ladder are making contact with the plasma membrane at regular intervals. So that led us to immediately ask the question of what's anchoring the ER rungs to the cell surface. And for that, we started focusing on this protein juxtaphilin, which is known to be an ER plasma membrane contact or tethering molecule. Juxtaphilin is an ER resident protein. It's got a transmembrane domain integrated into the ER, and then it reaches out and basically makes contact with the plasma membrane. Now, juxtaphilin is particularly interesting because when it is at the plasma membrane, it can interact with voltage-gated calcium channels, CAV, as an abbreviation. It can also, in the endoplasmic reticulum, integrate with calcium-dependent calcium release channels, in particular, the ryanodine receptor. So basically, what juxtaphilin is doing is bringing these two calcium-dependent systems together. So basically, when calcium crosses the plasma membrane into the cell as a result of the opening of these calcium-gated channels, a transient, the ryanodine receptors, if they are right next to there, will sense this calcium, open up, and then release calcium stores from the ER. And as a consequence, you basically get a localized hotspot of calcium at the sites where at the sites where the plasma membrane and the ER are being brought together through juxtaphilin. So we wanted to know whether this tripartite system of juxtaphilin, calcium voltage channels, and ryanodine receptor are regulating long-distance calcium signaling in dendrites. So for that, we started by just looking at juxtaphilin along the dendritic shaft of neurons. And surprisingly, we see a periodic pattern. So in purple, you're seeing juxtaphilin, and it's on the surface. You can see it lining up on the plasma membrane of that dendrite in a periodic way that matches where you're seeing in green, the endoplasmic reticulum is making contact with the plasma membrane. So you see the green ER ladder-like pattern. When it reaches the plasma membrane, it now co-localizes with juxtaphilin. We can also see that this anchoring, this juxtaphilin-based ER plasma membrane anchoring, is occurring all along the dendrite. And we furthermore can see that where the juxtaphilin is on that plasma membrane and these regular periodic arrays is where voltage-gated calcium channels are also localizing. This is shown in the fluorescent image to the right, where we're simultaneously looking at a calcium voltage channel with juxtaphilin. And you can see in the merged image that the periodic sites where the juxtaphilin is bringing the ER and plasma membrane together are sites where the calcium voltage channels are localized. We can also see that modulation of juxtaphilin levels can lead to a change in the organization of the calcium voltage channels on the plasma membrane. So if we reduce juxtaphilin levels, we get the calcium voltage channel dispersal. If we overexpress the juxtaphilin, we get more clustering of that juxtaphilin. So basically, the amount of juxtaphilin in these cells in the ER can lead to a change in the spatial organization of this calcium voltage channel from one that is dispersed to one that is highly concentrated. We can also see that the clustering of these juxtaphilin and calcium voltage channels can increase when neurons are activated, in this case with kinase acid. So this suggests that this is playing a very important role in some way in the way signals are moving within the system. Well, what about the ryanodine receptor, which is in the ER and it's this calcium-dependent calcium release channel that opens up in response to calcium? Is it also positioned at these juxtaphilin calcium voltage cluster sites? And the answer is yes. If we simultaneously express juxtaphilin, ryanodine receptor, and a calcium voltage channel in these dendrites, you can see they're all three of these guys co-localize at these clustered periodic arrays along the surface of that dendrite. So basically what our data is suggesting is as follows, that all along the dendritic shaft is an organization of machinery that involves calcium handling. The calcium voltage channels, the juxtaphilin, and the ryanodine receptors are localized in periodic arrays all along that surface. Well, what might that do? Well, it might lead to a mechanism, we think, for calcium uptake release hubs for long distance calcium signaling. So what do I mean by that? Basically, could stimulation of these dendrites enable a calcium signal to be propagated long distances because of this type of organization? So in order to address that, we did an experiment in collaboration with a group at Knox Point, Florida, where we did 2-photon uncaging of glutamate to excite single spines. We then monitored the loss of calcium from the ER through a reagent where basically an ER GCAMP molecule that we've targeted to the ER. And so the ER is green. And when we see release of calcium from the ER as a consequence of the ryanodine receptors opening in response to calcium signals, we should see a loss from that ER. And the question we want to know is, by locally stimulating this spine, can we see long distance signal propagation in the form of loss of calcium from the ER over long distances? And when we did that, we found the answer is yes. Basically, local spine activation drives a periodic calcium release from these ryanodine receptors at the ER plasma membrane junctions over microns, tens of microns. So the red arrow is where we stimulated the spine locally with a 2-photon microscope. We then basically, the green shows the pattern of loss of calcium from the ER as a result of the opening of these ryanodine receptors. If we inhibit the activity of the ryanodine receptors with ryanodine, you see it flat. There's no release from the ER. And consequently, no signal propagation of calcium from the ER. So basically what this data suggests is the following, that in response to glutamate, the NMDA receptors open, calcium goes in. AMPA receptors, which are also activated by glutamate, trigger a very rapid depolarization across that membrane. Now, what the depolarization does is open the calcium voltage channels. So you get a transient movement of calcium through these clustered arrays of calcium voltage channels into the cytoplasm. What does that do? Well, because the ryanodine receptors are positioned right next to these channels, you now open the ryanodine receptors and calcium gets released from the ER to lead to basically a calcium hub, increase in calcium that is distributed all along, will distribute all along that dendritic surface. So does anything special happen at these calcium hotspots that might be important for the way the neuron works? And the answer is yes. And a molecule called calmodulin kinase 2, we found gets recruited to these sites. This CAM kinase 2 plays a very important role in phosphorylating proteins that we know regulate synaptic plasticity and memory in the brain. And so what we're seeing is once you start triggering this system and you get these local hotspots of calcium along that dendritic surface, we get recruitment of CAM kinase 2, which is going to modulate these proteins all along that dendritic surface so that it can operate more effectively as a signaling machine. Here's the evidence for that. In the center is phosphorylated CAM kinase 2 that we're detecting with an antibody. When we don't have the cell activated by incubating in 5 millimolar potassium, you don't see accumulation of CAM kinase 2, even though we can see periodic pattern of the juxtaphilin and the CAM along the surface of the dendrite. When we activate that neuron with 90 millimolar potassium, you now see that the CAM kinase 2 starts accumulating. Phosphorylated CAM kinase 2 is accumulating at these periodic sites where the juxtaphilin and the ryanodine receptors are localized. That's significant because it implicates these calcium hubs that we're seeing and this calcium release and recycling machinery as crucial sites for modulation of proteins. If we block calcium voltage channels, we block the recruitment of the CAM kinase 2, the phosphorylated CAM kinase 2, consistent with basically this being a regulated system. So basically, what I've told you is a mechanism we think that's operating for long-range calcium signal integration in dendrites. Dendritic spine activation is going to trigger an initial depolarization across the plasma membrane that will lead to transient opening of the calcium voltage channels that are clustered along that surface. The input of calcium through those channels, those channels will trigger the opening of the ryanodine receptors from the ER that then release significant levels of calcium at those specific sites. We think that the individual sites of hubs, if you will, of calcium are potential, they're positioned potentially at distances that allow potential cross-activation and stimulation of these ryanodine receptors for a persistent calcium signaling at these sites, thereby allowing the recruitment of CAM kinase 2, its phosphorylation, phosphorylated forms of these calcium kinase 2, which then can directly impact, because these are kinases, they can phosphorylate proteins in the vicinity that then alter the characteristics of this dendritic system. So in this way, this system we think is operating as this long distance sort of communication system analogous to the cell phone towers that can take signals long distances throughout that dendritic system, allowing for super long communication from remote areas into the soma that then triggers an action potential that goes into tissues and other cells. So with that, I want to end. Thanking the people who made this work possible. In particular, I want to just sort of call out Lorena Benedini, who really drove this work, incredible postdoc. I also want to very much thank Tim Ryan at Bile Cornell, who basically co-mentored Lorena as a visiting scientist at Janelia. I also want to thank Pietro Giacomelli, who helped us conceptualize some of this, as well as David Klapman, who played a big role in our sort of ability to sort of think through all of these calcium-dependent processes. I also want to thank teams at Janelia that helped us with the electron microscopy, as well as some of the other two-photon work, in particular, Vidya Rangaju from Max Planck, Florida, who helped us do this two-photon work. Thank you so much. And if there's time, I can answer questions. Otherwise, thank you for your attention.
Hi, Jennifer. This is Win.
Hi, Win.
Thank you very much for a brilliant talk, as usual. So, two questions. In the past, there have been many studies regarding the effect of neural stimulation of the liver on many functions, which you summarized in your first slide. To my knowledge, despite asking this question many times, nobody has ever done any of those studies after a liver transplant. Because after transplantation in an animal or in humans, the vagus is no longer connected. And as a matter of fact, that whole parasympathetic stimulation is completely lost.
Yeah.
To my knowledge, nobody has ever looked as to what the downstream effects of that deinnervation is on any of the claimed metabolic regulation. The other thing is, if you speculate beyond the neural system, these connections exist in almost every cell, don't they? Between the ER and the plasma membrane. So what are your thoughts about the broader aspects of not just calcium signaling, but what do you think those connections mean in non-neural tissues? Because this seems to be a universal biologic effect.
Yeah, that's great. I'm going to focus in on the last question, which is, it is widely known that all cells, there is intimate contact between the ER and the plasma membrane for sort of communication pathways. In some cell types, however, this is much more significant for calcium regulation than others. Just as an example, in muscle, this system of juxtaphilin and ryanodine receptors are playing a very critical role for how muscle is able to regulate calcium-based contractile activity. So this whole pattern that you're seeing, that we're seeing in the neuron, is replicated in the muscle for regulating contractile across that whole huge muscle fiber. In other systems, and we're trying to investigate this right now, we're trying to see, what this kind of organization might play. Not clear, like in a HeLa cell or some other fibroblast or in a macrophage. Exactly. Certainly there's calcium signaling that's operating in this way, but because those cells are very small, it's not clear it's playing the equivalent function as we're seeing in these neurons or muscle systems that are huge, require huge communication across long distances.
Thank you very much again, Dr. Schwartz. Thanks for your time.
Yeah.
Now we'll move on to our next speaker, and it's my pleasure to introduce Dr. Sanford Simon. Dr. Sanford Simon is a Gunter Global Professor at Rockefeller University and a scientist who's renowned for how he has illuminated biology with studying one molecule at a time. And he is very well known for studying single channel transport to studying single viral dynamics. His work has repeatedly redefined how we visualize cellular events. Dr. Simon is not just an inspirational scientist, but he's also a very inspirational father. Where during his tenure as a scientist, when his 12-year-old daughter was diagnosed with fibrolamellar carcinoma, a pediatric cancer, he focused his research on studying about the fibrolamellar cancer. And from there on, he has pushed this understanding of this to a greater extent, and he has also established a big registry for, the big registry for fibrolamellar cancer. And he has been doing continuing research on these, and he has developed studies towards developing new therapies and repurposing drug for fibrolamellar cancer, and also developing new antisense and PROTAC strategies. With this introduction, we would like to welcome Dr. Simon, please.
So as a lab cell biologist, it's actually an honor to speak right after Jennifer. And Wynn, just thank you for an entire career of amazing stuff. And I'm still impressed with all the questions you asked. I actually have questions on your questions, but maybe over lunch. So I'm going to tell you how now a partnership between scientists, clinicians, and patients can actually work on addressing fundamental questions, both physiology, but also therapeutics and pathology. And I'm going to give you lessons from a particular rare disease, fibrolamellar carcinoma you just heard about. It was characterized in 56. Mainly, there were these very, very large hepatocytes. This is the tumor on the right, the adjacent normal from the same patient on the left. In addition to the large cells, there are these fibrotic bands made out of collagen. There's no established diagnosis. As a result, it's often misdiagnosed. Its peak detection is roughly 21. It hits kids roughly between 5 and 40 years old. It's a primary liver tumor. And unfortunately, there's very few symptoms, and often it's just not diagnosed until it's much too late, after it's already metastasized. It's still codified as a subvariant of hepatocellular carcinoma. And as a result, most patients are actually treated as if they have hepatocellular carcinoma, which is actually a terrible mistake. Unfortunately, no systemic chemoimmunotherapies have been found. So essentially, the only treatment available now is surgical resection. So when I started working on this, what was not known is this really sort of one disease or many that has similar phenotype was a genetically inherited or somatic mutation. So in 2008, my daughter was diagnosed with fibrolamellar. And the problems with studying a rare disease is that there's a lack of information, there's a lack of diagnostic tools, there's too few samples, and too little funding. But there are advantages to studying it. There are a number of rare diseases that people have studied because they're precisely defined. So for example, we've learned a lot about tumor suppressors studying retinoblastoma, which is an equally rare disease. So if they're extremely well characterized, you can take home lessons that you can then apply to many other aspects of physiology and pathology. So our approach was to partner with the patients through social media. We established a medical registry. We have data from over 270 patients, a tissue repository from over 160 patients. And through this registry, we made it as a nonprofit run by the patients. The reason being that there's a limited number of patients at any one institution, limited number of patients at any one hospital. By having the patients, the family members running this, we can then follow the patients over time as they go from hospital to hospital, as they go even across countries. And it means we can also update the records as time goes on. Rather than just being a registry where we follow the patients while they're in a particular clinical trial, we can follow them for the lifetime of the patient. Because it's being run by the patient's family members, we can guarantee that we will not sell the data to a private foundation, which gives us a tremendous buy-in on the part of the patients. And what we do for them, we provide information about the science. We're sort of like a two-way street with them. We have frequently asked questions about fibrolamellar that they can go through. We have dictionaries of key terms. We have a little video they can go, how to go into PubMed, how to search PubMed, how to get copies of the articles. We have plain language summaries of many of the articles on fibrolamellar. We've even made little videos on some of them.
In order to confirm that the chimera actually causes fibrolamellar, the researchers edited the genes of adult mice using the new CRISPR technology. They took the DNA of adult mice and created the same DNA deletion found.
So you can go into the video and follow, but the idea is to make the science accessible to the patients. We've moved the entire platform over to REDCap, and we have various people studying it. So, for example, we found that a lot of the patients are clustered very close to each other and not in population centers, which has led to a lot of interesting studies on some of the chemicals that could be driving this or triggering it. We found, for example, that women are more likely to get the disease. About 60% of the patients are women. But if you look at the survival, here in blue, women actually survive better than men. This has led to a whole study on sex-based differences between them. As we suspected, standard treatments do not work. Pretty much the only thing that shows any promise is surgery. As part of doing this, we found that most of our patients are told they're not candidates for surgery. And what we've been doing is encouraging them to get a second opinion. If you just look at the survival of patients who didn't go for a second opinion versus those who did, there's a big difference in the survival. Lately, we've started taking all the data that's been done on the characterization of the disease, and we've created a database that's accessible to the patients. We can go in and we explain to them what are transcriptomics, what are proteomics, phosphoproteomics, metabolomics. Why would it be of interest to them? So patients can actually go in and type in the name of any particular transcript they want. Like they're told, X, Y, or Z is increased in your body, and they can see how does it vary between the normal tissue, the primary tumor, recurrences, and metastases. Or for that matter, they can look at the same transcript and see how does it change in fibrolamellar, let's say, as contrast to hepatoblastoma, cholangiocarcinoma, hepatocellular carcinoma. And there are a lot of benefits back and forth for both the patients and the scientists. So as a result, many of our patients work at the bench, where they're actively involved in deciding a lot of our directions for therapeutics. Still more visit the lab. Many donate tissues. As I said, we have over 160 samples from different patients, and they share their medical records. As a result, they're very, very active in their participation. Our first clinical trial opened last May. It immediately filled up. Phase 1, phase 2 just started recently. So in doing our analysis of it, we did whole genome, transcriptome, proteomics, and phosphoproteomics on it. What we found was essentially there were very, very low level of single nucleotide variants of any mutations in the DNA. No obvious mutations in any of the known oncogenes, and a very low level of structural variants. So the question is, what's going on? A key came from doing the transcriptomics, looking at the RNA. We found 700 transcripts changed significantly in every patient we looked at. So if you look here at the transcriptome of the fibrolamellar patients, this is from 148 patients comparing the tumor to the adjacent normal. It clearly segregates away from the normal. In fact, the fibrolamellar transcriptome here in orange segregates away from cholangiocarcinoma, hepatoblastoma, or HCC. And here's the normal tissue. So it completely segregates away from other liver tumors as well. We found roughly 170 transcripts out of 55,000 differed in the metastases, of which only 39 were strongly consistent. We found the change of the proteome were pretty much the same patient to patient, the phosphoproteome and the metabolome. So the question is, what's going on in driving it? And one of the things we found was over a dozen kinases that were upregulated in every patient. And some are known oncokinases like RORA-A, EGF receptor, ERBB2, PRKCA. But they didn't have obvious mutations, so were they maybe using different splicings? Maybe that's what was going on here. So the approach we took was to take the RNA reads and map them onto the DNA test. Are they using different exons? We found for all the various oncogenes, for all the kinases, everything was normal except for the catalytic subunit of protein kinase A. So this is on the negative strand. So we're reading now from right to left. So here's the first exon, second exon, third. These are the reads between the exons that bridge them. When we compared the normal to the tumor, exons 2, 3, 4, they all increased in reads. The reads between the exons increased. The only one that didn't increase was the first exon, which was sort of strange, nor the reads between the first and second. Instead, we had reads that went upstream about 400 kilobases to the first exon of a heat shock protein, DNA JB1. And what this was suggesting is what we had was both complete reads for the DNA JB1, the heat shock protein, complete reads for this catalytic subunit protein kinase A, but now other reads that were the first exon of the heat shock protein followed by the catalytic subunit protein kinase A. When we went into the tissue from the patients, in every single patient, we found this chimeric RNA. We found a chimeric fusion protein that was made part of DNA JB1, part of PRKCA. In fact, when we walked along the DNA, every patient had one normal chromosome, but on one copy of chromosome 19, there was a deletion of roughly 400 kilobases, which could account for this fusion occurring. So this was found in every single patient we looked at, but it could be just an epiphenomenon. Is it really causally involved in driving it? So we went into mice using CRISPR-Cas9, created the same deletion, and found that we got cells that looked to the histopathologist, like fibrolamellar. The transcriptome was the same as what you see in patients, and they produced tumors, and the mice died from it. What this doesn't do is distinguish, is it formation of this fusion gene that's causing the cancer, or maybe it's the loss of one of the genes in between, maybe some kind of tumor suppressor. So to address that, we just expressed the fusion transcript by itself in mouse liver, and we found that was sufficient to produce the tumor. So these are the cells that were transduced. If we put in a point mutation that would kill the kinase activity and now no longer transduce, it means that not only is it expression of the fusion, you need the kinase activity as well to get transformations. Now the question is, how could this be driving it? So it turns out protein kinase A, unlike some other kinases, actually exists as a holoenzyme of two regulatory subunits. The regulatory subunits both inhibit the activity of the catalytic subunit and they localize it, keep it very restricted. It's only when you get a transient release of cyclic AMP that you release the catalytic subunits active and then it goes back. Maybe since this is now being driven by a heat shock protein, maybe we have more catalytic subunit. Maybe what you're getting is a lot of extra free catalytic subunit. So one possibility, you have more active kinase, or maybe if there's a real excess, it's changing its localization. It's no longer being restricted where it's found. An alternative possibility is just a change of the structure here of this fusion kinase, maybe affecting enzyme dynamics or interactions. We tried really hard to find any kind of difference between the fusion and wild type kinase, and they were the same under all the characters we looked. So is there a change in the amount of the catalytic to the regulatory subunit? Is there a change in the free catalytic activity and its localization? We found was that in every patient, there was a change in the amount of free catalytic subunit compared to the regulatory subunit. So on mass spec, if you just compare in the adjacent normal tissue here in black, the regulatory subunit to the catalytic, in every person, you've got more regulatory than catalytic in the normal tissue, and it regulates it. It keeps it inhibited. But if you look in the adjacent tumor, there's here in light gray, is the catalytic subunit. There's always an excess of catalytic subunit, suggesting it can't be inhibited, it can't be localized. We also did a variety of biochemical assays for it. We observed by confocal microscopy, proximity ligation. There's a lot of free catalytic subunit. If you just measure the basal catalytic activity, this is a log plot, normal tumor, there's an increase from 30 to 80-fold in the amount of catalytic subunit. That's the amount of catalytic activity in the tumor cells. Furthermore, if you ask where is the catalytic subunit, in most normal cells, it's in the cytosol. It's not in the nucleus. When you look at the tumor cells, there's an increase in catalytic subunit everywhere, but an incredibly pronounced increase in the nucleus of the cells. So these are just correlations. There's an increase in catalytic subunit, increased catalytic activity, change of its localization. But does it have a causal effect in driving the cancer? So the approach we took, and actually I should have pointed out all that previous work was the work of an amazing scientist in the lab, Masa Sharani. She took primary human hepatocytes and transduced them either just with a red fluorescent protein or a kinase dead version, or then compared it to just transducing them with the fusion kinase or just the wild type kinase. If it's really just an increased amount of catalytic subunit, that should be all you need. It also turns out that in certain pancreatic tumors and cholangiocarcinomas, there's overexpression of another fusion, ATP1B1 pukaka. All of these produce very, very similar changes in the transcriptome. You can see it better here. These are the changes of all the transcriptome from the fusion compared to the wild type kinase. So the changes in the transcriptome are almost the same. In fact, if you just express the wild type kinase, primary human hepatocytes, and put them back into the liver, that's enough to give you tumors that recapitulate the transcriptome of fibrolamellar. And in fact, if you compare the transcriptome of the cells we've transduced just for a week with those in the patient samples here in sort of yellowish orange, it's enough to recapitulate almost exactly the entire transcriptome of the patient tumors from just a few days of transcriptome of expression. So what do I mean almost? All of the classic transcripts were the same in the patient tumor as the cells we transduced, except for collagen. Every time we transduced an active subunit of the PK, the protein kinase A, and collagen went down. But collagen is always up in the patients. That's what makes these fiber spans. So the question becomes, why are they actually, decreased in these tumors? So why are they not increased? So we asked the question, where's collagen expression in these hepatocytes in the actual tumors? To be honest, if you look at the collagen expression during single cell spatial transcriptomics, the collagen expression is actually decreased in the tumor cells. It's increased in the surrounding stromal cells. So transducing the primary human hepatocytes with any of these actually completely recapitulate the actual transcriptome of the tumor cells, not of the tumor, because the tumor also has stromal cells in there. So it's suggesting that all you need is an increase in the catalytic subunit. So the question is there actual data in the patients to support that? So I mentioned that we had some patients that had tumors in the ductal cells of the liver, forming cholangiocarcinomas, or in the pancreas, that have a different fusion. But what does their transcriptome look like? So if you look at their transcriptome, even though this is a cholangiocarcinoma with a different fusion, its transcriptome is exactly the same as our hepatocytes with DNA JB1 Prokaka. In fact, if you do a drug response profile, if you look at the EC50 across hundreds of drugs, they have exactly the same drug response profile, even though there's a different fusion. So it's suggesting that all you really need there is the increase in catalytic subunit. It turned out we found 4 patients who did not have any fusion to protein kinase A. They looked like fibrolamellar to the pathologist. It turns out they were just missing the regulatory subunit. So instead of an increase in catalytic subunit, they had a decrease in the regulatory subunit. It was missing. Now, if you look at their transcriptome was exactly the same as the patients who had the fusion to PRKCA. So what this suggests is that this fusion domain is dispensable. All it's doing is giving you an increase of expression. All you need is increase the amount of catalytic subunit. So what can we do in terms of therapeutics? We tried everything that was clinically in use for in the clinic against fibrolamellar. None of them showed any therapeutic window. Well, what about things that hit protein kinase A? We tried dozens of drugs, and the problem is, while they killed the fibrolamellar cells, they also killed primary human hepatocytes. PK is an essential protein. But what if targets upregulated in fibrolamellar? We had identified aurora kinase A is upregulated, the EGF receptor pathway. Drugs against those showed no clear therapeutic window. Glutamate pathway was altered. The Wnt pathway was up. Whether we tested the cells isolated from patient-derived xenografts, fresh from patient resections, or even growing in mice, none of these drugs had any effect. But what about using antisense against the mRNA? The reason for doing that is that we want to know, is this DNA JB1 Prokaka fusion a useful therapeutic target? What I've shown you so far is just that it's triggering the disease, not that it's continuing to drive the disease. So the question is, if you get rid of it, do the tumors just keep growing? And for that matter, do they just become normal, or are they actually addicted to it? Do they die? So we tiled across the junction, came up with some shRNA that would eliminate the fusion protein, but not the wild type protein. If we have tumors growing in mice, if we induced it, so here's an inset here. When you induce expression of the shRNA against the fusion, the tumors not only stop growing, but they die, which tells us it's not only triggering the disease, it's continuing to drive it, and they're oncogenically addicted to it. We've recently started using siRNA as well. They also stop growing. They also start to decrease. There's some problems with delivery, but I'll go into in a moment how we're getting around that. So antisense against the RNA works. What about coming up with degraders? And so our approach was the following. This is the native kinase on the right. Here's the fusion kinase. The native kinase is tightly folded. It's not a good substrate for the proteasome. We asked, what if we actually target something into this active site of the kinase here? By targeting there, that means that the kinase cannot mutate to escape it. And now, what if we now put a little extension on it so it's linked to an E3 ligase? If we now express this degrader, we can now selectively get rid of the fusion protein, not the wild type. You see the tumors are growing, but if you turn on this degrader, they stop growing. There are a lot of potential concerns. They could overexpress the kinase. So if we just normally have this kinase here, we can get rid of it. When they're overexpressing it, they're still degrading it, but they can't overcome the overexpression. So we've started doing now is instead of just having our targeting peptide in E3, we're now having, as part of the same mRNA construct, an antisense. What we do now is we now degrade this much faster, much deeper. And lately, so this is the work of Massa, who also did the pathogenesis. Shaq, who's been working on the combo, and DeVere now is working on targeting it. By targeting lipid nanoparticles to deliver the mRNA, we can completely get rid of the fusion protein and kill the tumors. So the thing is, people are dying now. What can be brought to the clinic now? So we did a drug repurposing screen against 7,000 compounds. And what we found was 3 classes of compounds showed efficacy. Irinotecan, which should work against rapidly growing tumors. This is a slow-growing tumor. Inhibitors of HDAC work, like panobinostat, and inhibitors of the anti-apoptotic protein BCLXL. I got worried because none of these made sense to me in terms of the biology. So I thought maybe this is an aberration of working on patient-derived xenografts in mice. So we shifted to working on tumors directly from patients and found that all 3 of these compounds worked on tumors directly from patients. But there was variability in the response. They all responded to irinotecan. Here in black is the primary hepatocytes. But what was the source of the variability? So I thought, well, each time it's a different tumor. We're resecting it for we're digesting it for different periods. Maybe we're mishandling it. But what we found was we had some patients where we had samples years apart, and samples taken years apart showed exactly the same drug response profile. In this case, these are 3 Mets compared to a primary 3 years earlier. In fact, you looked at all the drugs, they responded the same within a single patient. The difference patient to patient was in the level of expression of that anti-apoptotic protein I mentioned, BCL-XL. Just what if we combine inhibitors of let's say, irinotecan with blocking BCL-XL? So this is the patient who's least responsive to irinotecan. When we put in increasing amounts of drugs to block BCL-XL, we can now drive the sensitivity to below single-digit nanomole. We worked on a degrader that will get rid of BCL-XL. And now if we have tumors growing in mice, when we just treat with irinotecan, the tumor stops growing. When you stop the treatment, it doesn't come back even for months later. We mix it with a degrader for BCL-XL, and it goes away and doesn't come back. This degrader is now being tested as monotherapy for other purposes. From going through our registry, we found 9 patients who had received it, and in fact, the tumor stopped growing. And so we opened up a clinical trial on this back in May. Phase 1 is completed, phase 2 has just started. But what worried me is like, why is irinotecan working? It should only affect rapidly growing tumors. So irinotecan is made into SN38, which enters the liver, where in the liver, this family of enzymes puts on a sugar group to export it out to get rid of it from the body. It turns out the increased levels of protein kinase A downregulate the promoter in this in such a way that this is now down about 50, 60-fold, and that's why they're sensitive. Panobinostat, the HDAC inhibitor, is actually metabolized by CYP3A4, which is down 1,000-fold in fibrolamellar. So why is fibrolamellar a good system for cancer? It's extremely well characterized. While there's a different DNA break in every patient, the RNA is the same, so the protein's the same. What works on one should work on all. We can show a fusion oncoprotein sufficient to create it. It drives the tumor. It's oncogenically addicted to it. And there's a real need. Drugs in the clinic have no effect. Drugs like pathways are upregulated have no effect. But functional precision medicine can have an effect. And therapeutics against the fusion oncoprotein, this antisense degraders, are potential therapeutics that could apply to many other cancers and diseases. So since the patients did the bulk of the work, I actually want to thank the patients. This is my daughter who initially found the deletion and the fusion and actually did all the early transcriptome and proteomics. Jackson did the early protein purification. Jackson passed away right after his 26th birthday. Mark, also 26. Willow did our organize, passed away at 21. Tani, Anna, passed away at 20. These patients really drive the work. I just want to thank the members of my lab who've been absolutely great. And thank you for your attention.
Thank you very much, Dr. Simon. This is truly an inspirational and example of a translational work for us. We have time for one question and more during the chat session, so we can have like one-on-one discussion during the chat.
I was wondering, this is really a beautiful presentation, and congratulations on your efforts in getting together the patients and the families. I was wondering if the metastatic disease in these patients displays any additional mutations or changes in transcriptome.
So we've examined the metastases from patients. There are 39 transcripts that change significantly. There are no new mutations in them. Their drug response profile is exactly the same, whether it's a MET or a primary. We found 39 transcripts that change reproducibly, but it turns out almost all those changes are due to changes of the stromal cells at the bulk transcriptomics. When you do single cell spatial transcriptomics, we think we have it down to maybe just 3 or 4 transcripts that are different, and we don't know if those are significant yet.
And the other question is, when you have effect of irinotecan, so would you give that as a sort of preventive medication for patients who potentially could have micrometastasis?
That's a tough question that we debate in the lab, particularly with the patients. We're pushing very hard now to develop assays to see are there micrometastases. We're working on circulating tumor DNA. We now have particular cell surface markers that we're now using to try to create imaging agents to see if there are micrometastases. So far, with a combination of irinotecan and DT2216, we don't see any adverse effects. I'm just nervous about giving that to people. We're pushing very hard on our combination of degrader and antisense, where we've shown, at least in mice, we can massively deliver it with absolutely no change in physiology. I think I'd feel more comfortable just... Okay, this sounds like a non-professional evaluation, but in every case, the standard I ask is, would I give this to my daughter? And maybe that's not the proper way clinically to decide, but that's how I make my decision.
In a larger sense, what your work illustrates is the critical importance of having bridges between patients, clinicians, scientists, because at the present time, for the most part, those are almost like separate disciplines that don't really communicate very extensively with one another, other than maybe sharing tissue or something like that. But the input that you get by bringing patients, particularly those with rare diseases, together with scientists, with clinicians, is really the avenue of great hope for the future, rather than, this is a personal opinion, rather than just having people operate in their own silos. This kind of bridging is what's, I think, desperately needed in much of our research. So thank you.
I think right now, in many ways, we're in this glory age of science. And if you look at what's been done in recent years, like you mentioned earlier, hepatitis C, the SARS-CoV-2 vaccine. But I think we as scientists have failed to convey that to the public. I think by having the patients, instead of asking patients, oh, give us your tissue, participate in the trial, but actually engaging them in it so they really see what's going on. I think that's what's critical of developing an appreciation for what we're doing. So we actually give seminars and get involved with patients all over the world. And I'm amazed at how many of these patients and their family members come from political backgrounds where they just never appreciated what's actually going on in science. I think Francis Collins described it. What you want to do is not just invite them, the patients, to the table, but actually bring them into the kitchen, have them prepare the menu and prepare the food.
So we'll quickly move on to the next exciting part of the session. So we have the Microtalk speakers. So we have 3 speakers in the first session. So the first person is Daniel Schaefer from Gössling lab at MGH. Daniel Schaefer.
Hi, thank you for the introduction. As was said, I'm Dan Schaefer, a computational technician in the Gössling lab, and today I'm going to give a brief overview of an autoimmune hepatitis project I have been a part of. AIH is a rare, poorly understood liver disease characterized by abnormal liver tests, suggestive histopathology, and steroid responsiveness. There are currently no diagnostic criteria that are both specific and sensitive for AIH. In search of an AIH biomarker, we performed single nucleus sequencing on tissue from 32 human livers. We analyzed data from both diseased and normal controls, with disease controls including MASH, steatosis, and PBC, as well as an unknown sample and 2 overlap AIH PBC samples. The diagram shows that our disease cohort consisted of patients who were considered probable AIH pre-biopsy, and our normal tissue was gathered from a cohort of patients who were undergoing surgical tumor resection. Single nucleus RNA sequencing is a method that measures gene expression at a single cell resolution, but only measures the RNA in the nucleus. Using gene expression, we can identify and quantify cell types in each sample. On the right is a visual representation of the cells we captured, where each dot is a cell and different colors represent different major cell types. When normalized to the total number of nuclei in the sample, we get a fractional abundance value for each cell type. Our main analysis was centered around how the abundances of different cell types varied between diseases. These same colors represent the cell types in the abundance bars on the bottom, where the height of the bar represents the relative abundance of a cell type in that sample. Already we can see that disease samples have a different cell type abundance profile compared to normal samples. We were interested in whether the cell type abundance data alone was enough to distinguish AIH samples from the control samples. For each sample, we combined the cell type abundances of major subtypes and minor immune subtypes and performed naive clustering on the resulting vector. Across 3 different methods, with the third shown on our poster, we consistently saw the AIH samples clustering separately from the control samples. In the plot on the right, I show 2 clustering methods where the disease labels were not provided. On the y-axis is each sample label with a color representing the patient's final diagnosis. 2 shades of blue represent AIH patients. On the x-axis is the cluster that is sorted into. We can see that AIH separates well into its own group, apart from other diseases and normal samples. This is really exciting because it suggests that cell type abundance distributions alone are enough to distinguish AIH. I would love to share with you the cell types driving this distinction, as well as the beginnings of an experimental validation in my poster. Thank you.
So next speaker is Fabian Surup. Fabian.
Hi, everyone. Let's get this a little bit higher. My name is Fabian Surup. I'm a fourth-year PhD student in Anna Greca's lab here in the Broad. And I want to briefly introduce to you how we can leverage shared, or what we call nodal biology, to solve some of the unmet needs in rare diseases. We just had a great example earlier of the challenges in the therapeutic development for rare liver diseases. And so when we think of unmet need in our society, we often think of the most pressing concerns of being, for example, Alzheimer's disease, which affects 7 million people, or perhaps cancer, affecting 18 million people in the US. However, an even greater cumulative unmet medical need is that of rare diseases, affecting over 30 million people in the US. However, as rare diseases are individually rare, the therapeutic development for them often remains insufficient. In our lab, we're interested in using nodal biology, that is shared biology across different genetic diseases, to accelerate the therapeutic of new drugs to access ultimately more patients. We've been able to identify a group of cargo receptors called TMAT cargo receptors, which contribute to the accumulation of misfolded proteins in disease, and have been able to show that when we target these, we're able to achieve clearance of toxic misfolded proteins and the aggregates in genetic diseases of the eye, kidneys, and brain. Now, as today's focus of the symposiums of the liver, I want to present this paradigm in the context of the most common genetic protein accumulation disease of the liver, namely alpha-1 antitrypsin deficiency. In alpha-1 antitrypsin deficiency, variants lead the protein to misfold, polymerize, and accumulate within hepatocytes, ultimately leading to cellular stress and chronic liver disease. To closely mimic the human disease biology, we partnered with a group at Boston University to obtain patient iPSCs that were able to differentiate into hepatocyte-like cells. These cells accurately lead to accumulation of misfolded protein as identifiable by the green signal in these cells. We're then able to treat these with a small molecule drug that leads to the degradation of our nodal biological targets of the TMAT cargo receptors, and observe a potent reduction of the misfolded protein, as you see here in the absence of the green signal. When we quantify this, we also observe that this is almost at the baseline background staining of a wild-type patient control. Including this data and other data I haven't presented here, we were able to identify that at baseline, in the presence of TMAT cargo receptors, large ATZ polymeric aggregates are insufficiently degraded, leading to disease. However, when we target TMAT cargo receptors, either genetically or by pharmacological perturbation, we're able to accelerate lysosomal degradation of these toxic aggregates and are likely able to restore disease pathology, which we're currently testing in animal models. So here briefly, we've just shown how we can use nodal biology to alter rare genetic liver disease biology. Thank you. My poster is number 20, way in the back, so come find me there.
Our next speaker is Florian Hügeler from Macromolecular Engineering Lab. Florian.
Hello, everyone. It's a great pleasure to introduce you today to a new treatment for non-alcoholic fatty liver disease instead of hepatitis. And before, I would just like to quickly establish the therapeutic need that we actually have, because as many of you know, there are only very few treatment options that we can give to patients with fatty liver disease. And so many of the patients actually face a transition of the disease towards later stages, including cirrhosis and even liver cancer, which ultimately requires a liver transplant. On the other hand, if you look on the transplant side, many of the grafts that are donated today are unfortunately also from donors with fatty liver disease. And so these grafts cannot be transplanted because they're prone to dysfunction after transplantation. And so many of these grafts are discarded despite the global need for transplants and the global organ shortage. And so we had the goal to actually come up with a new treatment, a pharmacological approach that we can give on one hand to patients with fatty liver disease, as well as to actually recover additional grafts for transplantation by actually treating grafts that are donated outside of the body before implanting them into patients. And so our approach was to actually use PI4KB inhibitors. And I'm not going to go too much into the details here, but you're very welcome to join me my poster 11, where I have mapped out the whole mechanism of action. But basically, we treated this first and tried it in mice that were given 24 weeks of a high-fat, high-fructose diet to develop steatohepatitis. And then they were treated for 12 weeks with these PI4KB inhibitors. On the other hand, we also wanted to try this ex situ, so outside of the body. And therefore, we wanted to leverage this perfusion platform which is essentially just the pump that oxygenizes blood or perfuse it, pumps it through the liver, and can keep up human livers for up to 2 weeks outside of the body in a viable and functional way. And so we were able to test this with a discarded human liver graft that we received from Swiss transplant, which was steatotic and had a high amount of fat in it that we then could use to actually try the treatment in vivo. So when we look at the results, we can actually see that there is a quite remarkable effect of the drug, because when we look already at the H&E staining, we can see a clear reduction of lipid droplets. Same also goes for the oil red O staining, where we see in red the lipid droplets as well. And then even when you look at the trichrome staining, there is an improvement for the fibrosis that we can see. So when we then look also at blood values, so for example, graft inflammation, we can see that there is a clear reduction also of inflammation as reflected in these transaminases AST and ALT. And then, of course, there's a clear reduction in tissue triglycerides. When we then tried this in the human liver graft XC2, we can see that there is a clear trend of reduction of tissue triglycerides already in just 8 days of perfusion. And so we're quite hopeful that this would work also in human patients, because we could confirm the mechanism of action. And so in the very sense of this meeting, we've done our basic science now, so we'd be very excited to actually find someone who's interested to try this in patients at some point. Thank you very much.
Thank you. Thanks for all the speakers. And so this gets to the, a break session for the first part of the morning. So we'd like to apologize for all the Zoom questions because we were not able to address them during this first part. So we'll definitely try to do our best to cover the Zoom questions in the second half of the session.
It is my honor to introduce Dr. Irwin Arias, a distinguished leader in liver biology and medicine and a rare triple emeritus professor of medicine at Albert Einstein College of Medicine, professor of physiology and medicine at Tufts University School of Medicine, and senior scientist at the NIH, where he continues to teach. Dr. Arias has spent his remarkable career linking basic biology to human disease, contributing to groundbreaking discoveries in inheritable jaundice, bile transport, and the regulation of hepatocyte polarization, work that reshaped our understanding of liver disease. A founder of the American Liver Foundation and a mentor to generations of scientists and physicians, he continues to inspire us. Please join me in welcoming Dr. Irwin Arias.
So first of all, thank you all for being here, and I hope you enjoy this as much as Luba and I do. We learn an enormous amount from these bridging activities. So, quo vadis bile acids? Because I suspect that most of you, particularly the younger students here, are not highly informed about this area of bile acid biology. So my brief presentation is really going to be almost introductory to the outstanding work that you're going to hear about, or we're going to hear about in the next speaker. And I guess it was Newton who said that, the great advances are made by people who are standing on the shoulders of visionaries of the past. And bile acid biology is a wonderful example of this. And it shows how you reach a point in research and understanding where you think you understand everything, but we never do. And this is a beautiful example of new doors opening almost with every generation. So we go way back to antiquity, when both the Greeks and the Romans knew there was something magic in treating people who had arthritic pains, digestive symptoms, didn't feel well in their head, and they were treated with bile acids. And interestingly enough, we'll come back to this at the end of this, is that the bile acid that had the best effect was from bears. Well, now we know that's ursodeoxycholic acid. But there were other biles, and you can go to a restaurant in Canton, I think it still exists, many of them, where snake biles are the cure-all. But it wasn't until the 20th century in Europe when a whole group of outstanding chemists got to work and actually worked out the structure and the many different types of bile acids. And there were extensive studies of different bile acids in different species. And there's a whole litany of this, which were summarized in a magnificent collection about 15 years ago by the late Alan Hoffman, who published it. Now, along that time, physiologists came up with the important discovery that bile acids are derived from cholesterol. Well, it was known that bile acids are detergents. So here nature is converting a detergent into essentially deriving it from cholesterol, using the formation of a detergent as a way of getting rid of excess cholesterol in the body, which, as we all know, is not good for you. And to show you how important this was, there are 6 Nobel Prizes that were given for this work in chemistry and in the relationship to cholesterol. The physiologists also discovered that bile acids, to a great extent, the primary ones made in the liver, are recycled back from the intestine. They are secreted, they facilitate fat absorption and emulsification and so forth, and then they're reabsorbed, usually in the terminal ileum, so there's this enterohepatic circulation. And early studies were done where perfused liver was then subjected to various inhibitors of energy, and it came to the conclusion that there was active transport involved in secreting these things. And it was blamed at that time on the sodium pump. Weak evidence, because the sodium pump generates about minus 35 millivolts, which was never enough to achieve the high concentration gradients that one found of bile acids. But nobody paid much attention to that, because in those days, ATP was used for ions. It wasn't used for organic molecules. And so the function was simply that the detergent emulsified fat and promoted absorption, and it was related to a lot of diseases. Now, at that point, the biophysicists kicked in, and particularly Don Small here at Boston University. Don used to quote Caesar, all Gaul is divided into 3 parts, but Don's 3 parts were cholesterol, bile acid, and phospholipid. And the point was that these 3 come together to form a micelle. And there's a critical micellar concentration, which is required to get rid of cholesterol, which is in the center of that micelle. And so they measured the critical micellar concentration and found in people with cholesterol gallstones that that concentration was out of whack, and you could restore it by feeding a bile acid. They initially gave kino, and actually cholesterol stones dissolved. Took time, and if you stopped taking the bile acid, they were returned. Well, that study, which was monumental, took a good number of years to perform, and by the time they had finished it, laparoscopic surgery had come into being. So by laparoscopic surgery now, of course, surgeons don't have to give general anesthesia. They operate without opening up the abdomen, and that basically replaced the use of bile acids in most instances for the control of cholesterol gallstones, which could be removed laparoscopically. And then it took about another 50 years before the cell biologist and the transport people got in the act. And part of it was the discovery of a family of ATP-dependent transporters located in the apical domain, the biocanalicular domain of the hepatocyte. We actually contributed the first of those. And one of them, ABCB11, is specific for bile acids. And the proof of the pudding was the discovery of young children who had recessively inherited defects, molecular defects, in ABCB11, producing progressive familial intrahepatic cholestasis type 2. And this is a significant disease. It's rare. But it proved the point that this is the major way that the liver gets rid of bile acids, by transporting it, not through the sodium pump, but through an active ATPase. And also a bunch of other transporters, importers and exporters, were found in the basolateral part of the cell. And it is argued that they protect the organelles of the hepatocyte from the detergent effect of a bile acid. And they do to a limited degree. But in most forms of bile secretory failure, called cholestasis, bile acid accumulates within the hepatocyte, and it raises hell with every membrane that it contacts, most notably the mitochondrial membrane. And so mitochondrial failure, which has been dissected to an increasing degree, is an essential part of the secretory failure that results in the whole complex diseases called bile secretory disease or cholestasis. And then a new era emerged, and that was the impact of molecular biology, with the discovery that bile acids are actually signaling molecules, and they are controlled and control an FXR nuclear receptor system. And so the emphasis has entirely shifted, or did shift for a great degree, time to the role of bile acids in terms of their control of various signaling dependent pathways, largely through the FXR regulatory complex. Now, at this time, bile acids were known also to increase bile flow. And one of the common human diseases, primary biliary cirrhosis, is a disease of the small bile ducts. And it was argued that they had a decreased flow, and therefore, if they were treated with a bile acid, things would get better. Well, they did get better. And it was initially in that clinical trial that suddenly the floodgates opened. So where are the floodgates? In today's literature, there is evidence and research of bile acids having a role in inflammation, in immunology, in the microbiome, in development, in the formation of a polarized epithelium in the hepatocyte, and in metabolism, in a wide variety of hormone actions, most recently the glucocorticoid receptor, and even in regeneration. And there have been claims in transformation. What else? Well, this is a floodgate. And the hard evidence in support of any of these is only slowly being accumulated. And we're going to hear next a talk by Siva Varanasi, who is a new appointment at the University of Massachusetts Medical Center, where he's an assistant professor. He received his PhD from the University of Tennessee and trained in his home country of India, in Hyderabad, and at the Vellore Institute of Technology before he came to the United States. He published this year an outstanding paper on the role of bile acids in regulating immune responses. And some of this came from his work at the Salk Institute, where he worked with Ron Evans and the group who played a critical role in the discovery of the FXR regulatory complexes. So it's a great pleasure to introduce him, and we certainly look forward to your presentation, which is entitled Bile Acids as the Master Regulators of Immunity in the Liver. So, Dr. Irwin.
Let me take a moment to actually enjoy this, because I could not get a better introduction in my life than from the legend, Win himself. So I'm truly honored. Again, I'm truly honored to present here, but also get such a phenomenal introduction from Win about bile acid. That cut shorts a lot of my talk. But our lab works on immune system in the liver, mainly this type of immune cells called T cells, which you must have all heard of. These are adaptive immune cells that control intracellular pathogens and tumors. T cells actually play both pathogenic and protective role depending on the disease type. And for example, like in viral hepatitis, T cells are dysfunctional or so-called exhausted. In autoimmune hepatitis, T cells are more damaging, tissue damaging. And we have other examples like NASH and liver cancer, where the same effect of protective versus pathogenic roles of T cells play. So our lab, we are interested in understanding how the metabolic state of the liver modulates T cell differentiation, because you get these different T cell subsets in the liver, and the metabolic properties of the liver change with different diseases. So what is the connection between those two? And can we modulate the liver metabolism to regulate T cell responses? Or can we engineer T cells metabolically to make these T cells either better fit or not, to treat the liver diseases? I think that is the overarching goal of my lab, and I'm going to show you some directions that we're taking. Like Win mentioned, we focused on bile acids, for many reasons. One of the things is, it gives this tissue-specific properties. If you look at T cells, entering all these tissues, we wanted to understand how they survive in those tissues. Bile acids are these tissue-specific metabolites that are made by the liver themselves. And they're also actually dysregulated during multiple liver diseases. So that fits the criteria of how they could influence T cells. And more importantly, they are also shown to be involved in other diseases beyond liver, like to the extent where people have shown that people with psychiatric diseases or depression actually have dysregulated bile acids, that adding to the list of things that Win has shown, where all the floodgates have opened for bile acids. So bile acids have this both systemic and also tissue intrinsic effects, given with the liver diseases aspect. For example, we know in NASH patients or cirrhotic patients, people show higher bile acids in their periphery. We know with alcoholic hepatitis, that is the case. The bile acid levels go up in the system. And we recently showed that in the liver tumors too, that you see an increased bile acid levels within the liver and also in the periphery. So the question we asked is, in the lab, is how does bile acid modulate distinct T cell responses? Because like I mentioned, there's three disease models here. NASH, where T cells are pathogenic, they cause the tissue damage. On the other side of the spectrum, you have liver cancer, where T cells are dysfunctional. So how does bile acids modulate T cell responses differently? To understand how bile acids affect T cell responses differently, we need to understand how T cells differentiate differently. You could get different flavors of T cells depending on the disease context. The three major flavors are, you could get resident memory T cells upon acute infection, for example, listeria infection or a plasmodium infection that is cleared. So you could generate these memory T cells in the liver that are long-lived, and so they are functional T cells. You get these hyperactive T cells. These are tissue-damaging pathogenic T cells that are constantly damaging hepatocytes or killing hepatocytes and other neighboring cells in the liver. So these are hyperactive T cells, and they are wired differently. And you get these dysfunctional cells, aka exhausted T cells. These are T cells that are that have disability in their function while still present in the tumors or in the livers. So we see that in hepatitis B and C infection. We see that in hepatocellular carcinoma infection. So you generate all these different kinds of flavors of T cells, and now you add bile acids, around them. What effects do you get, and how do they shape these responses? So we're gonna break this into different type of T cell differentiation. So I'm going to first talk about T cell exhaustion or dysfunction and its role of bile acids in shaping those responses, and then go down the route. So to test how bile acids would shape T cell dysfunction in the liver, we used the liver cancer model that is primarily driven by SV40. SV40 is a viral oncogene. It's not really, expressed in the liver. But we use this model to actually, for two reasons. One is it drives tumors in the organ that you express. You could express it in any organ. It'll lead to tumor. The other beautiful reason is that it is processed by the hepatocytes and presented, and you could transfer transgenic T cells that recognize the immunodominant epitope of SV40 and track those T cells. So it gives us this tumor-specific T cell responses, so you could track, as opposed to studying bystander T cells, T cells that are just flooding into the liver as a consequence of inflammation. So you could really distinguish between what is an antigenic T cell or tumor antigen T cell versus not. So the first, the basic experiment is you transfer these transgenic T cells that recognize SV40. You see this classical signs of exhaustion where T cells are expanded initially and then they're crashed. They express, a lot of different cytokines, for example, interferon gamma initially, and then they lose their function. So this model, is a great model to study T cell exhaustion in the liver. And we then looked at bile acid changes in the liver. We saw that induction of liver tumors, just like in human patients, we see a robust induction of primary bile acid synthesis, as exemplified here by the expansion of at least 10 to 50-fold of certain bile acid species. Most of these bile acids are primary bile acids made by the liver themselves or hepatocytes themselves. We do see some changes in the secondary bile acids that are made by the microbiome. I'm not going to talk about that today, but I'm happy to chat. So we see this increase, in this, at least 10 to 50-fold of bile acids in the liver. So we asked, is this mere tissue damage that you see during liver cancer, where you see like AST, ALT levels go up, the bile acids are going up because of the tissue damage, or is it actually contributing to the cancer? So we then used this AAV approach, where we added a guide RNA cassette to the AAV TBG-CRE, which actually is used to turn on the oncogene. So we then knocked out every single gene in the bile acid synthesis pathway and then asked the question, which gene is important or which pathway is important for regulating tumor burden and also T cell responses? We actually were surprised to see this result, because when we knocked out each of the enzyme in the bile acid synthesis pathway, until knocking down BAT, which is the enzyme that's involved in the conjugation of bile acids, just reminding you that 95% of all bile acids made by the liver gets conjugated. So none of these other enzymes actually made much difference. Like if you knock out CYP7A1, you still get tumors. CYP7B1 and 8B1 would still give you tumors. It's the knocking down of all the conjugated bile acids by knocking down BAT would actually gives rise to this strong effect on anti-tumor properties. So where we see a robust reduction in tumor burden, these mice respond to immune checkpoint blockade. Otherwise, normally liver tumors, both in patients and also mouse models, don't respond to immune checkpoint blockade, and that is another key bottleneck in terms of immunotherapy. So the surprising fact was that, as I mentioned, the T cell responses die off by 2 to 3 weeks after you transfer. However, in the BAT knockout mice, or when you knock out BAT in the tumors, you actually see sustained T cell responses to the tumors. And this was surprising because liver is such a harsh environment for T cells. Even with just anti-PD-1 would not give rise to a robust T cell response. So we were getting such a strong T cell response by manipulating bile acid synthesis. We also see other changes when we did single-cell RNA-seq, like, for example, changes in macrophages and some extent, in CD4 T cells. But we see this, induction of this tumor-reactive T cell expansion in the liver. So we then asked a question. This is a classic experiment. What Win would do, if he had a lab, is if you culture T cells in vitro and then add bile acids to them, would the T cells be affected with bile acids? So we did the exact experiment where we cultured in vitro activated CD8 T cells and added all the different kind of bile acids we could purchase, from Sigma. And we saw two major effects. One is an effect on survival, and one is an effect on function. And those two are independent. For example, the primary conjugated bile acid, the TCDCA, or chenodeoxycholic acid version of it, was actually killing T cells when you are in a dose-dependent manner. Whereas the inhibition, the dysfunctional effect that we see, where we see loss of T cell function, was actually primarily derived by the gut microbiome-derived bile acids. These are different forms of LCA. It didn't matter which LCA it was. All the LCA species were inhibiting T cell function. We saw an increase in T cell function with UDCA, like Win mentioned, a bare bile acid, was actually pro-inflammatory to T cells. So this effect of survival and function was really uncoupled, depending on the different bile acids. So TCDCA was killing T cells, LCA was inhibiting T cell function. So we looked at Win's old papers and looked at, okay, what causes T cells to take up bile acids and either have it or release it. I think his classic papers on P glycoprotein or MDR1 was really inspiring. So we wanted to look at that a little bit more. But we first asked this question, whether T cells in the tumor take up bile acids or not. So what we did was we FACS sorted tumor-reactive T cells and the bystander T cells from the liver tumor and performed mass spec, just to see whether T cells in the tumors would take up bile acids or accumulate bile acids. And we saw actually that tumor-reactive T cells would accumulate TCDCA, the one that is killing T cells in vitro. So that was, again, supportive to our hypothesis that T cells were taking bile acids up. We performed an RNA-seq comparing those bystander T cells to tumor-reactive T cells. We found that the bystander T cells were in fact expressing the efflux transporters like ABCB1 or MDR1 or P-glycoprotein, which is important for effluxing bile acids, whereas the exhausted T cells or the tumor-reactive T cells were not expressing that transporters. So something within that tumor-reactive T cells is inhibiting for these cells to express these transporters. In fact, that is the case in human TILs too. If you look at human TILs that are these antigen reactive or PD-1 positive, lion positive T cells, they actually express less of these efflux transporters, SLCO3A1 and to some extent ABCB1. So we did a couple of experiments. We knew that ABCB1 was important. Work from Mark Sunderd's lab was really a pioneer in establishing that, where they knock out ABCB1 and T cells crash in vivo. We found a new transporter called SLCO3A1 to be also important and also regulate bile acid efflux. When we knock out SLCO3A1, T cells also don't persist in the liver tumors. When we overexpress these transporters, it's a sufficiency experiment to see whether that is enough to actually cause T cells to persist in the livers. We see exactly the same thing. The overexpression of this isoform 1 for this efflux transporter was enough to promote or boost their survival in the tumors. So again, suggesting that efflux transporters play an important role in T cell survival in the tumors, which is primarily affecting the tumor-reactive T cells for some reason. I'll get to the reason, in a second. Is that all the case? Is that enough, to explain the bat knockout phenotype? Not really, because one of the things that also happened in the bat knockout, or when we knocked down bat, is we saw an increase in this bile acid called UDCA. bare bile acid, like Win explained. So we were pretty surprised to see a consistent increase in the UDCA levels in the bat knockout. Now we made a bat knockout mice. We actually see the same thing. The UDCA levels go phenomenally up. So something is shaping the microbiome to make more of these UDCA. We fed the mice with UDCA. This is 4 weeks after tumor initiation. The window of tumor initiation is only 6 weeks. So in the last 2 weeks, literally, we fed the mice with UDCA and saw a dramatic effect on reduction in the tumor burden. This was phenomenal, again, suggesting that UDCA could be repurposed, for other diseases, beyond cholangitis, which is now prescribed, in clinic. So we could actually try some clinical trials on UDCA for liver cancer. That's my hope. So what I told you in terms of cancer is that there are two arms of bile acids affecting T cells. One arm is the hepatocyte-derived bile acids killing T cells because T cells don't express or express less of the efflux transporters. These are tumor-reactive T cells. And so as a consequence, T cells take up bile acids, would cause mitochondrial toxicity, like Win also described. What would happen when a cell would accumulate bile acids? As a consequence, you get cell death. The other axis is the microbiome-derived bile acids, where microbiome is changed. They make a lot of LCA species. These LCA species would cause, ER stress in T cells and would cause T cell dysfunction. We also see that there's other bile acid receptors T cells will express. I can talk about that, as a last future direction slide. So this is all in the tumor-reactive T cells where T cells are dysfunctional, and this would explain that. Now, where are we going with, to understand why would T cells be hyperactivated in other diseases like NASH or autoimmune hepatitis, for example? So that's where we're going next. So to study the autoimmune hepatitis, we used this phenomenal mouse model from Anne Rothstein's lab, who's next door, at my institute, where she found that actually mice lacking DNase 2, which is important for cleaving DNA, leads to systemic inflammation and interferonopathy. And the mice actually die in utero. So you have to cross it to interferon alpha receptor for these mice to survive longer. So what happens in those mice is as they grow older, they accumulate a lot of these immune cells or T cells primarily near the portal vein, and that leads to fibrosis and causes autoimmune hepatitis or chronic inflammation in the liver. So this DNase 2 knockout model actually presents a great tool for us to understand how autoimmune bile acids would shape T cell responses in autoimmune hepatitis. So the first thing we did was we measured bile acid changes. To our surprise, actually, we saw huge amounts of bile acids in their serum of these mice, almost to 100-fold increase in bile acids. Not only primary conjugated bile acids go up, but also unconjugated bile acids also go up. So that is something really interesting about this inflammation, inflammatory model. So a talented grad student in the lab, did some single cell RNA-seq analysis as well as spectral flow, and actually found that these autoimmune hepatitis mice actually have these population of T cells that are expressing characteristics of a dysfunctional cell, like what you would see in an exhausted T cells in a tumor, for example. They would express PD-1, they'd express TOX, they would express CXCR6, and all the phenotypic markers of an exhausted T cells. But in fact, they actually are highly cytotoxic. They express high levels of granzyme B and TNF alpha and interferon gamma. So we coined this term hyperactive T cells, just for the ease of it. So he then did a single cell RNA and TCR-seq on these livers and actually found that these mice that show signs of autoimmune hepatitis actually have this hyperactivated T cell population, this PD-1 positive CD4, CD8 T cells, while there's other CD4 T cells that are also expanded. But these PD-1 positive CD8 T cells are hyperfunctional. Like I mentioned, they express a lot of gamma and granzyme B. Because we did TCR-seq, we actually saw that these T cells actually are clonally expanded. A lot of them are, are also present in the spleen. So there is some systemic effect of these, of these knockout mice. So the T cells are coming in clonally expanded, coming into the liver and causing tissue damage. So if you look at the top 10 clones, in these livers that are expanded in these mice, we actually find that each of these clone is unique to itself. It's not like all of these are expressing all the cytokines. Actually, for example, clonotype 1 would not express, granzyme A would express high levels of granzyme K. Normally, you don't see granzyme K expression in T cells from the tumor or in the memory T cells. It's a unique granzymes that is expressed in the autoimmune, diseases, not just liver, but also other tissues like autoimmune, encephalitis, and also Crohn's disease. So granzyme K is expressed in certain clonotypes, granzyme A is expressed in certain clonotypes, and interferon gamma is expressed in certain clonotypes. So each of these clonotypes were unique, in that sense. So we asked, where are they located? What's happening, in these livers? So we then measured, the location, of these T cells in the liver, and actually saw that the enzyme CYP7A1, which Win mentioned, is the enzyme that is the first enzyme in the bile acid synthesis pathway, which is normally expressed by the central hepatocytes, the hepatocytes that are close to central vein. That the expression of that is completely destroyed. You could see a destroyed, zonation in the liver and associated with T cell accumulation, mainly in the portal regions of the liver. So you could see this formation of these triads where Kupffer cells, CD4 and CD8 T cells come together and form these triads. That you could see that, readily in this, in these autoimmune mice. So what we then did was this, this brave action, because we did single-cell RNA and TCR-seq from autoimmune hepatitis model. We have a single-cell RNA and TCR-seq from a liver tumor model. We combine those two together and ask the question, how do T cells differentiate differently? What are the different characteristics of T cells? So this is what I'm showing you. We combined these data sets together more recently and actually found these three subsets, like I mentioned. There are these resident memory T cells population here that are present in the livers during both liver tumors and also during autoimmune hepatitis. There is exhausted T cells that are, primarily present in the tumor mice. There are hyperactivated T cells that express granzyme K. These are primarily present in the autoimmune hepatitis mice or in this, in this DNase2 knockout mice. You can see that the PD-1 expression marks, most of these hyperactivated and exhausted T cells. So we then asked, what are the different what comes out of these differential expression analysis? And the top gene that came out from differential expression analysis is the efflux transporters. So it turns out that the hyperactivated T cells have no problem expressing efflux transporters, whereas the exhausted T cells actually do have that problem. So there's something about the differentiation of these exhausted T cells that is inhibiting their expression of these efflux transporters, both ABCB1, which is MDR1 or P-glycoprotein 1, or SLCO3A1, which we study to be important also for bile acid efflux. So we confirmed that. So we took these two mice together, we isolated T cells from the livers, and then did an efflux assay where we added this rhodamine dye and then asked how much of this dye gets effluxed out when you incubate them in the incubator. The hyperactivated T cells actually efflux out majority of this dye within 30 minutes after incubation, whereas the exhausted T cells have the dysfunction in effluxing this particular dye. Again, confirming the expression analysis that the exhausted T cells express less of this efflux transporter. So where are we going with this, or in our lab, is that we think that bile acids affect T cells, or actually different kinds of T cells, differently. They modulate ER stress, they modulate mitochondrial stress through efflux-dependent mechanisms. They also modulate bile acid receptor signaling. I haven't told you about bile acid receptor due to time, but I'm happy to chat. But each of these different signals actually drive distinct fate in T cells. You could get memory, hyperactive, and exhausted T cells depending on what signals are generated, what bile acids are present in the environment. So we are trying to integrate all these signals together and study T cell fate in the liver during liver diseases. That's the goal. So with that, I'd like to conclude my presentation and thank my lab, Tim, who did majority of the work, Sophia, who's also in the audience, who can chat, Sushmita, who did a lot of the seq analysis, and Rothstein's lab, Karen from her lab, helped with a lot of these DKO mice and autoimmune hepatitis models, and our metabolomic score. My postdoctoral mentor, Susan Keck, who actually gave me that foundation to start working on bile acids in T cells, and American Liver Foundation, who actually believed in me and gave my first grant. It was quite a depressing year with everything happening, but American Liver Foundation was supporting this work and funded me for this work. So I'm really glad to have received that. It kind of boosts your morale. So with that, I'd like to take any questions.
Are you concerned that mice, their primary bile acids are hydrophilic rather than hydrophobic? Muricholate. So my question is, you're studying tumors in mouse livers and studying the T cell relationship to mammalian bile acids, which are only expressed to a very limited degree in the rodent. The rodent doesn't have that kind of a bile acid. So are there comparable effects with the muricholate and the hydrophilic bile acid, not the hydrophobic ones?
That's a great point. Actually, our first experiment in the lab was to knock out the enzyme CYP2C70, which is involved in the murocholic acids, in the rodents. And we actually saw more aggravated tumors. So the murocholic acid turned out to be more, I'd say, more calmer bile acids, like trying to protect, the livers from getting tumors. So the CYP2C70 knockout mice would get more aggressive tumors. In terms of direct effects on T cells per se, we did see some effects with the murocholic acids on T cells. We tend to not go in that direction just to make sure that we are focused on the bile acids that are primarily expressed in humans, the taurocholic acid and the glycocholic acid, glycoquinodeoxycholic acid. So we tend to stay within the human bile acid profiles more, rather than focusing on the murine-specific ones. But yeah, there are interesting things happening on the murocholic acid side as well.
That was a beautiful talk for any year, regardless of funding agencies and their issues. But I was wondering, how do you think the bile acids are getting into the T cells? And is that related to these differential effects that you see on different bile acids, or do you think, or are there differences in the complement of bile acid receptors like FXR, TGR5, et cetera?
Yeah, I want to defer that question to Win, but actually I'll try to answer, because I did spend almost a year, actually wasted my whole year trying to figure out the transporters for bile acids in T cells. What are the importers? Because T cells don't express classical bile acid importers like SLC10A1 and other, like if you knock out those receptors and, bile acid would still cause an effect on T cells. So there's two possibilities. One is the passive, infiltration or not passive influx of bile acids, independent of transporters. The other hypothesis is that there's transporters in T cells that we don't know of. There's a lot of these orphan bile acid receptors that we don't know of, like SLC03A1 was one of that for a long time. So somebody has to come up with the transporters, and then we can find out in T cells. But so I couldn't find out. Yeah, thank you.
I have a couple of questions, if I can. Great talk. I'd like to ask whether you see any difference in the sensitivity of T cells to bile acids. So you've done differentiation in terms of function, but say CD4 versus CD8, do they respond differently? Because I'm sure you are aware of effects of bile acid on Th17 cells and so on. So this is the first question. Second question is, do you know what upregulates your ABCB1? There are reports that say hypoxia-inducible factor 1 alpha can induce ABC transporters. So I want to know your view if you have evidence.
So you want a sneak peek. Regarding the first question, yes, there are different effects of bile acids on different cell type. I think there's like beautiful papers in Nature in 2024 talking about some of the microbiome-derived bile acids like LCA. We talked about how it has pro-Treg promoting functions inhibiting Th17 cells. Through different mechanisms, they look at NR4A1 and ROR gamma T. So these bile acids that we talked about in CD8 T cells, the LCA ones, they have a different effects in Treg. So that is one question. In terms of what drives the expression of these efflux transporters, there's a couple of evidences, one from our lab and one from a lab that's a Beth McCormick lab who's at also UMass Med, where they're looking at microbiome-derived changes, microbiome bile acids, driving those expression of ABCB1s. In our case, we think it's the cytokine milieu that the T cells are differentiated in. For example, type 1 interferons is one of the cytokines that we thought was driving. So we recently found that actually IL-27 is involved in driving the expression. So if you differentiate T cells in the presence of IL-27, you increase ABCB1s. So that was one signal.
Thank you. In the interest of time, we will take one last Zoom question, and then please ask your questions during the lunch break.
We have multiple Zoom questions. I'll just pick on one question and ask here. So it's from Dr. Jim Boyer. The question is, are bile acids getting into T cells via a specific transporter? If so, which one, as opposed to interacting with bile acid receptors like TGR5?
Yes, the actual importer is not known from to my knowledge. But in terms of bile acid receptors, I think that's a different question. Except for TGR5, which is expressed on the surface, most of the other bile acid receptors are intracellular, mostly the nuclear hormone receptors like FXR. There's some evidence that there's CAR also is involved, VDR. But we found that actually those bile acid receptors are important in T cells. Like when you mess their function or activate them, they do cause a distinct effect. But whether that explains bile acid import into T cells is not I'm not entirely sure. So to answer that question, I don't know what is importing or what transporter is required for bile acids to get into the cell. This is mainly a problem for conjugated bile acids, though. Unconjugated bile acids, like secondary bile acids, don't need a transporter. They could get into the get through the membrane easily. Thank you.
Thank you for that wonderful talk, Dr. Varanasi. Next, it is my pleasure to introduce Dr. Ellen Roche, a leading innovator in cardiac device engineering. Dr. Roche directs the Therapeutic Technology Design and Development Lab at MIT, where her team pioneers cutting-edge approaches to cardiac device design. Dr. Roche received her PhD at Harvard University, training with Professors David Mooney and Connor Walsh. She has also worked in the medical device industry with Mednova, Abbott Vascular, and Medtronic, and her groundbreaking work includes the development of a biorobotic hybrid heart, an image-based biomimicry that combines organic tissue with synthetic soft robotic matrices, work published in Science Robotics. Today, Dr. Roche is going to talk about hemodynamics in Fontan-associated liver disease. Please join me in welcoming Dr. Ellen Roche.
Hi, good morning, everyone. It's a pleasure to be here. I feel grossly underqualified to be here because I'm definitely not a liver expert, but I'm excited to talk for the next 20 minutes about how we think about linking hemodynamics in a type of congenital heart defect with a liver disease called Fontan-associated liver disease. I mentioned I'll talk about this congenital heart defect. It's called single ventricle disease, and it's when children are born with one ventricle or pumping chamber in the heart instead of two. They typically undergo 3 surgeries before the age of 5. In which their heart and the vasculature is reconstructed. So they have a very different hemodynamic physiology compared to biventricular physiology. There's over 1,000 Fontan surgeries. The last surgery is called the Fontan procedure, and there's over 1,000 per year done here in the US. There's over 70,000 people living after this Fontan surgery globally. And this is increasing. It's due to double in the next 20 years. I'm fascinated by this circulation because instead of two pumps in parallel, these patients live with one pump, which pumps systemically. And then the return to the lungs is passive. The venous return to the lungs is passive through a surgically implanted shunt or conduit. And because of this, there's really interesting interactions between the cardiac hemodynamics and respiratory effects. So during inspiration, as shown on, maybe you can see my cursor here, as shown here, when somebody inspires, the pressure in the thorax decreases and the abdomen increases, and you get this kind of surge of venous return to the lungs. But conversely, during expiration, when the thoracic pressure increases and the abdominal pressure decreases, you get this downward or retrograde flow, which goes to the abdominal organs, including the liver. And we became really interested in studying this retrograde flow in these patients and how it affects their prognosis and specifically how it links to liver disease. So Fontan-associated liver disease is a big problem in these 70,000 people living with the Fontan physiology. As you can see here, the surgically reconstructed shunt returning blood to the lungs is passive, and you get this reverse flow going down to the liver through the hepatic vein. And we've become really interested in studying that in a variety of ways in the lab, constructing physical models to understand it better and doing clinical imaging studies at Boston Children's Hospital in patients with this physiology. So in this Fontan pathway, there's no subpulmonary ventricle. There's a passive graft that's usually made of Gore-Tex material inserted to link the vena cava to the pulmonary vessels, to the lungs. It leads to complications, and about 40% of Fontan patients, they get Fontan failure by the age of 40. Over 60% need intervention within 20 years of their Fontan procedure. And a lot of this is driven by the adverse venous flows that I described and the cardiorespiratory interactions. So as you can see here, as the patients inspire and expire, you get this switching of flow to the lungs and downwards into the abdominal organs. So first of all, we started to try to understand this a little better by building physical simulators of the Fontan physiology in the lab. So we can use robotic techniques to build an overall cardiorespiratory simulator that's shown on the bottom here. We used artificial muscles to move a diaphragm as it would move during inspiration and expiration. And we had a flow loop with reconstructed patient-specific vasculature. And in there, we put a soft robotic ventricle, so we can use these artificial muscles to pump this ventricle at the heart rate. And now we can instrument this with flow probes and pressure probes and really understand the interactions between the breathing mechanics and the flow in this population. And using this, we can adjust things like rate of breathing and depth of breathing, and we can have the simulator be upright or down and really understand how to kind of tweak these interactions. We then worked with a team at Boston Children's Hospital to look at whether we could understand better the cardiorespiratory interactions in patients. So we set up this MRI study with Fontan patients. We've done about 38 to date, and we're really here trying to understand hemodynamics and how we might screen patients and understand who's at risk of potentially progressing to Fontan-associated liver disease. So we had patients do breathing exercises within the MRI scanner. We had a respiratory protocol where we had this app. You might know the meditation app on your watch or on your phone. And this helped us to encourage patients to adjust their breathing rate and to go from slow to fast breathing. We also had them breathe shallow and deep breaths to vary intensity. And then we designed this MRI-compatible ergometer so that they could exercise within the scanner. So we could see how different levels of exercise change these cardiorespiratory interactions. Then we used MRI to image about 2 millimeters above the hepatic vein so that we could quantify the flow in the vein when they do these different breathing exercises. So we found strong correlations between increasing respiratory effort. Here, patients go from shallow to deep breathing, so very shallow breaths to deep breaths, and these are measured by a chest belt. They're maintaining their respiratory rate constant, and we found a big increase in the retrograde flow to the abdominal organs, including the liver, when they breathe deeper. Similarly, when they maintain the same respiratory effort but increase respiratory rate, so they go from slow to fast breathing, we see an increase in the retrograde flow. So just these non-invasive breathing interventions where they're changing the intensity and the rate of their breathing has a big impact on the flow to the liver. Then we had them exercise, and we've been really privileged to work with these patients who volunteer their time after their routine MRI to do these exercises with us in the MRI. And we had them exercise at 3 different cadences of pedaling, and we can vary the resistance on the ergometer too, so the different levels of exercise. And we showed a decrease actually in the reverse flow. So exercising more changes the flow to the abdominal organs. So this is exciting, but we don't want to always have people in an MRI scanner to understand these. So we're now looking at, can we screen at-risk patients with ultrasound? So what we've started to do is compare the MRI-based ground truth study to an ultrasound or echo-based study. And if this can work, we can look at patients in the supine and upright position, and we can have them do different types of exercise and really understand in more detail these interactions. So for the ultrasound study, we have the same respiratory protocol, the same exercise protocol, but now we're looking at using ultrasound, and we're specifically interested in imaging flow in the hepatic vein. So here's what we can see, and this is with the color Doppler ultrasound. So this color map here shows forward and reverse velocities. And you can see this is the inferior vena cava here and the hepatic vein here. And we can track throughout a breathing cycle how this changes. So this is one example. But what you can see, as we showed in the MRI, as you increase respiratory effort, during exhalation, you see this reversal in flow. So the color changes here and you get this reversal of flow. Similarly, at increased respiratory rates, you get this flow reversal here. So even with color Doppler, you can see this effect. And if we quantify the velocity over time as patients inspire, inhale, and exhale, the reverse flow in the hepatic vein is actually even greater than that in the IVC. So there is this big hemodynamic effect, and we think that it's very much linked to Fontan-associated liver disease. So we wanted to kind of explore that correlation a little bit more. So we used a FibroScan controlled attenuation parameter studies, and we looked at patients who had done this exercise study where we had data on their hemodynamics and to see if we could correlate that with some liver scans. So here we're looking at controlled attenuation parameter, which measures the amount of fat in the liver. We can also measure stiffness with a FibroScan. In Fontan patients, actually, they tend to have higher stiffness even if they don't have Fontan-associated liver disease. So the absolute values here aren't as useful, but the trends are longitudinally. We've just done this for 7 patients so far, where we've taken FibroScan data from 7 patients, where we've also done the hemodynamic studies for, and we're trying to correlate the retrograde or reverse flow ratio with the FibroScan data. And we see a very strong correlation so far. This is very early data, but we see that the more retrograde flow that the patients have, shown on the x-axis here, the higher their CAP score. So this really suggests a link between Fontan retrograde flow and liver steatosis. So this is super interesting to us. Of course, we are device designers and we want to think about a way that we can prevent this reverse flow. Obviously, the natural inclination is to put a valve in the conduit. Okay, so we've simulated how we can insert a minimally invasive valve into the conduit and what type of valve design that we would need to achieve this kind of prevention of reverse flow. And we've done computational studies on that. We can also use an extravascular valve to prevent reverse flow in the conduit. One solution that we've been working on recently is a cuff that goes around the outside of the Fontan graft. What we see, the benefits of this is to have a sphincter that doesn't contact blood, that goes around the graft to prevent reverse flow. And we see that this could be self-powered. So it could harness the breathing mechanics to shift fluid from a pressure harvesting bladder to the cuff so that it would be synchronized with breathing and also not require any external hardware. So this would be respiration paced. And if I play this video, we've tested this in the lab as well. So when a patient would breathe, the cuff would close during expiration and prevent reverse flow. And we've shown that we can achieve this cessation of retrograde flow on the bench. We're excited about this because it's fully implantable. There would be no driveline that could lead to infections. It would be less likely to cause clotting because it's extravascular, no blood contact. And it would be compatible with current grafts, so it wouldn't change the surgical procedure that much. So we're building on the learnings to date with the physical robotic models and the clinical MRI and ultrasound studies, but we still really want to understand why this reversal in flow is leading to Fontan-associated liver disease. So we're excited recently to have been awarded a HEALS grant with Sangeeta, where we will take the learnings from our clinical and computational models and apply them to liver-on-chip models. So if we can use patient-specific or kind of disease-specific hemodynamic parameters like flow and percentage of retrograde flow and pressure, and apply them to these liver-on-chip models that have been developed in Sangeeta's lab, potentially we can really understand the mechanisms a little bit more. Then we can use computational models and our physical models to assess patients and know when to change. There's fenestrations from the Fontan shunt to the heart, and they can be closed over time. And if we understand better when they should be closed and when we should intervene and change the hemodynamics in these patients with these patient-specific models, we can treat them and avoid them having to have a large repeat intervention. So with that, I want to thank the wider team. This is a team of engineers here at MIT and in my lab, and a lot of clinical partners over at Boston Children's Hospital who have been amazing to work with. And thank you all, and I'm happy to take any questions.
My understanding is that almost all patients who had this surgery end up developing some amount of fibrosis, and that happens many years later. What are some of the early predictors that can help predict how soon it would develop? And the second question is, how effective are breathing exercises or just exercising good at managing the diseases currently?
Yeah, thanks, Fabian. Great questions. I don't think there is a great way to predict who's going to develop this liver disease at the moment or how quickly they will. But we're hoping that if we can, create these models where we link the hemodynamics to liver disease, we can screen earlier because these patients all have routine imaging and ultrasound for as part of their checkups. And if we can integrate kind of longer imaging of their flow patterns, and maybe that can help us to do earlier screening of who might be at risk. And then regarding the breathing exercise, this is something that we have done recently, and the surgeons at Boston Children's Hospital are excited to potentially have a way to modulate the flow non-invasively. So some of the next steps from that work are to partner with the exercise lab at Boston Children's Hospital, where they have these patients do different types of exercise. They have some data on what helps in terms of exercise, but we really want to be able to image during different types of exercise what's happening in the hemodynamics. And maybe we can come up with a better exercise regime or protocol to help these patients and potentially prevent progression to liver disease. But it's very new at the moment. There isn't a great understanding of it. So hopefully we'll be able to help create these protocols. Thank you.
Ellen, could you clarify a little bit the 65% roughly of Fontan surgical patients you said require revision in 20 years? Now, I may be wrong, but my understanding is the revisions are brought about because they really develop, it's almost like a Budd-Chiari kind of fibrotic liver disease. And that's the problem, that they develop liver failure.
Yeah.
So what is the revision?
Yeah.
And is that a liver transplant, or what are they revising? And the other thing is, could you amplify a little bit what you meant by fenestra?
Oh, fenestrations.
Effects.
Yeah, absolutely. Yeah. I can start with the first one. So yeah, if it's liver failure, it would be a liver transplant. But Fontan failure is generally the revision there would be often they'll increase the size of the Fontan conduit to change the hemodynamics, or potentially they'll do a full heart transplant. So some reintervention is typically required within 20 years, which is much longer than it was previously. These people are living longer, and some of the patients we meet are very healthy in their 40s. But yeah, depending on what the disease is, it could be liver transplant or increase in the Fontan conduit size or heart transplant. To the second question, yeah, the Fontan fenestrations. So sometimes these conduits are put in with a branch to the heart, like an unloading fenestration. So it's a small little channel from the graft to, say, the atrium. And that can be closed over time and often is to modulate the hemodynamics. But knowing when to do that is something that, without computational modeling and advanced imaging, it's hard to know when is the best time to do it. They do that minimally invasively with small occluder devices in the fenestration or the branch. But to predict when is the best time to do that is difficult, because as the child grows, their hemodynamics change, and at some point you want to close that branch.
So we have two online questions. So one is from Don Rocky. Fascinating talk and lovely work. I have always wondered whether the injury is shear or simple pressure. Any thoughts?
Shear or pressure? Yeah, exactly. And that's part of this work where we're collaborating with Sangeeta on the liver and the chip, is to modulate the flow parameters to understand, is it shear, is it pressure, or is it this flow reversal? I think we want to, systematically vary these parameters and understand what's causing the disease.
So the physiology of severe tricuspid insufficiency from rheumatic fever prior to cardiac surgery results in cardiac cirrhosis. This would seem to be what occurs over time with Fontan surgery.
Potentially, this flow reversal, it could be a similar mechanism. And some of these valves that we're working on for the Fontan physiology can be used for other conditions to prevent flow reversal in the same way. Yeah.
Amazing work. I was very interested in your CAP data, and there seems to be increased hepatic steatosis. Do you think that it is a contributor to Fontan hepatopathy?
I think that's what we're going to figure out. This is early. We have just 7 patients that we have like this FibroScan data and the hemodynamic data for. But I think it could be. So we want to learn more about it. It definitely seems to correlate with these flow patterns. So yeah, we're going to include longitudinal FibroScan imaging in these patients as we monitor them over time. Thank you.
That was a wonderful talk. Thank you, Dr. Ellen Roche. Next, we have the much awaited Microtalks from trainees. Can I ask all the trainees for the Microtalk speakers to come near the stage, please? And first, we have Kahia Liu from Boston Children's from Bidinger Lab.
Hello, everyone. My name is Kahia Liu from the Bidinger Lab at Boston Children's Hospital. Today, I'm going to show you some interesting changes in your hepatocyte lysosome when you have Thanksgiving dinner for too many days. As we all know, lysosomes are conventionally thought to be the garbage bins in a cell. However, in metabolic happy organs such as liver, lysosomes also perform a role of serving as a central hub for lipid handling. Shown here is the sort of our current understanding of how lysosomes how this role is performed by lysosome. Lipids, as shown here, lipids contain rich in lysosomes through receptor-mediated endocytosis. And then in the lysosomes, lipids, for example, cholesterol, are further metabolized and then redistributed to other organelles or membranes. Given the global burden of overnutrition-driven metabolic syndrome, we wanted to understand what happens to hepatocyte lysosomes when you feed mice a short-term diet rich in burgers and fries. Shown here are the lysosomes in green in the control liver. As you can see, they're tiny and randomly dispersed as described in the textbook. However, under dietary stress, as you can see, these lysosomes became huge, sometimes even bigger than half the size of a hepatocyte nucleus. Biochemical assay and lipid profiling told us that these lysosomes under dietary stress are enriched in lipids and have a preference towards cholesterol. And quantification told us that dietary stress significantly shifts the size distribution of lysosome towards the larger aspect. And similar phenotype we're seeing in the context of lysosome storage disease, where these lysosomes are filled with lipids, eventually become dysfunctional and contribute to liver fibrosis and liver failure. In addition to size, we're also interested in the overall lysosome distribution under dietary stress. As shown here, the lysosomes the organization of lysosomes seem to be changed under dietary stress. This becomes more evident when you zoomed in. As shown here, under dietary stress, these lysosomes are redistributed to the certain part of hepatocyte membrane labeled in red here. Thanks to Dr. Arias' work, we now know that hepatocytes are polarized cell. With basolateral, to simplify, the basolateral membrane of hepatocytes are the nutrient inlets, whereas the apical membrane of hepatocytes are the outlets of bile salts and cholesterol. Surprisingly, we found that under dietary stress, these lysosomes were redistributed to the apical membrane of hepatocytes, which is further quantified here. Together with the other data that I showed on my poster, which is poster 16, we came up with a model where we think there might be an alternative route for excess cholesterol handling by hepatocyte lysosomes. In conjunction with the canonical roles, under dietary stress, these lysosomes might also traffic to the apical membrane and may directly dump the excess cholesterol into the bile. This seems to be a smart way and low burden way of getting rid of excess cholesterol because once in bile, the next destination of cholesterol is to get secreted out of the body through feces. And we're currently working on testing this model in in vitro systems. So if you have any ideas or comments, please come talk to me. My poster is 16. Thank you.
Thank you, Dr. Liu. Next, we have Brittany Tian from UToronto Ogawa Lab.
Okay, so thank you so much for inviting me here. And I actually came from Toronto, Canada. I'm not a local. And I will be pitching my project on making a bioengineered 3D HPSC cholangiocyte duct to model biliary disease. For our lab, we have developed a model that is more representative of intrahepatic bile ducts as opposed to like a lot of extrahepatic bile duct liver model that you probably know, like with bioengineered organ on chips and all of that. So we have collaborated with Dr. Boyoung Zhang from McMaster University, also in Canada, and he developed this 384 well system that is fairly high throughput, and then you can make a tubular structure in the middle well, so that's shown in that like that hydrogel structure there, and then it's a fully epithelialized bile duct, and we switch the direction on a rocker and put the plate on a rocker so that will give the bidirectional fluid flow. And here are some confocal images to show you just how this duct look like. So it is actually very representative of how a human intrahepatic bile duct is like physiologically. So you've got all of the key functional characteristics, including apical polarization of the primary cilia, and that is one single cytoplasmic protrusion towards the apical membrane. So you can see those green structures is protruding at the apical site. And then also CFTR, which you might know from the cystic fibrosis disease. That's also a very important transporter that can become dysregulated in biliary diseases, and that is also polarized apically. And I want to show you this video. A lot of people say that it looks like Mario Kart. So this is if you walk through the lumen, it pretend that you're a bile acid. Yeah. So what we can do with this model is that we can do a lot. So we want to model physiological signals, right? And that includes fluid flow, biliary stromal cells, bile acids, and this is trying to integrate all of the important signals. And a lot of existing models, they don't do this. Maybe it's just one signal at a time, or like they don't integrate everything comprehensively. And that's what my poster will be about. All of the data is in my poster, and it's like all the different bile acids, the major ones they can find in the human body, and as well as some inflammatory cytokines, even combinatorial effects between inflammation and bile acids. And as well as fibrosis modeling. So if you're interested, you can come by the poster number 5 during the lunch. And yeah, what we want to do with this system in the future is iPSC-derived cholangiocyte therapy, and also you can use this system to comprehensively study like mechanisms, disease mechanisms, and also disease modeling. So thank you so much.
Thank you, Dr. Tian. Next, we have Vardhman Kumar from the Bhatia lab.
Hi, everyone. I'm Vardhman, and I'm excited to be presenting my work on injected self-assembled image-guided tissue ensembles. To give a brief background, more than 2 million people die of liver diseases each year, and liver transplant is really the only option for end-stage liver diseases. However, it's limited by a shortage of donor organs worldwide. Clinically, hepatocyte transplantation has been tried as an alternative strategy, but the engraftment of the cells has been really poor. To address this, we and others have developed the concept of an engineered graft that can sit ectopically and provide the function of the liver. However, most of these approaches are procedures that require surgical interventions. To address this, we decided to work on an approach in which we can have the tissue be engineered in a way that's injectable. The cells can stay localized, become vascularized, and accelerate engraftment. For this, we turned our attention to a novel class of biomaterials called granular hydrogels, where instead of having a bulk hydrogel, the scaffold is instead made of these building blocks that come together and form the construct. We used step emulsification method and gelatin methacrylate hydrogel to create microspheres from this hydrogel in a high throughput manner. And the properties of this material when packed together is shear thinning, wherein when force is applied, it can be extruded out of a syringe, but when the force is stopped, it behaves as a solid material. Once injected, it maintains porosity because of the void spaces that exist in the interparticle region. When implanted in vivo, you can see in the case of bulk hydrogel, the blood vessels form a clear interface between the hydrogel and the vessels, versus in the case of granular hydrogels, which is composed of these microspheres, the blood vessels are able to penetrate through the void spaces, thus accelerating vascularization of the scaffold. To incorporate primary human hepatocytes within this culture to engineer liver tissues, we first aggregated primary human hepatocytes with supporting fibroblasts and created a slurry which consists of these microparticles and the primary human hepatocyte aggregates. In vitro, they showed sustained function over a period of at least 2 weeks, where the incorporation of supporting stromal cells was helpful at maintaining and enhancing their function. In vivo, for hepatocyte transplant, we decided to test 3 conditions: hepatocytes transplanted on their own in an ectopic space, or with supporting stromal cells, or with supporting stromal cells in the presence of these microspheres. And we do this under the guidance of ultrasound so that we can see how they are localized. In the first video, which is showing injection in the absence of microspheres, you see a bolus appearing, but it quickly disperses throughout and it does not stay localized. Versus when injected in the presence of microspheres, they stay at the injection site and the cells are localized within that. And because of distinguishable contrast between these microspheres and the surrounding graft, we can also monitor the shape, volume of this graft in a non-invasive manner over a period of time. Histologically, at the end of 8 weeks, we see findings that corroborate our observations in the ultrasound injection, where in the conditions where we did not have microspheres, the cells appear as discrete nodes spread and dispersed throughout the tissue, versus in the case of microspheres, we usually get a single tissue-like structure where the cells are dispersed only in the regions between the microgels, and the tissue remains confined at the injection site. And functionally, this condition outperformed the conditions where we did not have microspheres incorporated. Further characterization shows that the hepatocytes that are engrafted sit in close proximity to the mouse vasculature and are also polarized, as shown by bile canaliculi staining in the explants. Finally, we decided to change the material properties of these microspheres to have them be degradable. So once they are injected, then they go away while just leaving the hepatocytes to engraft. And we see that there's a decrease in graft volume because of the degradation of these hydrogels over time. And histologically, also, we can see that the microspheres have degraded over time, allowed for the graft to become more vascularized, which is also reflected in enhanced function of these grafts. And in our ongoing work, we are looking at using these microspheres as carriers for cargo to deliver therapeutics that can further increase the graft function. We're also looking at developing other long-term non-invasive methods for tracking the cells once they have engrafted and optimizing location of graft delivery. With that, I would like to thank Sangeeta, collaborators, and the funding agencies that have supported this work. And I invite you to my poster number 2 for more granular details. Thank you.
Thank you, Dr. Kumar. And with this, I would like to conclude the morning session. And thank you, everyone, for staying on time. And please return promptly for Dr. Gordon's talk at 1:30 p.m. Thank you.
Good afternoon. We're here for the second session. All right. My name is Dilnar. I'll be a session chair with Alex. Please join me in welcoming Dr. Arpan Mohanty. She is a hepatologist and clinical researcher at Boston Medical Center, an assistant professor of medicine at Boston University School of Medicine, where she also directs clinical research in gastroenterology, and she's a current recipient of the Robert Wynn Career Development Award and also serves as a principal investigator on multiple liver disease trials at BU. So please join me in welcoming Dr. Mohanty.
Good afternoon, everyone, and thank you for the kind invitation and the incredible honor to speak to you about my topic today, which is which I have named Unseen, Undiagnosed, Finding Liver Fibrosis in the Community. This is very different from the talks that you've heard. It's really clinical, and I hope I can engage you on this. So today I'll be talking to you about metabolic dysfunction-associated steatotic liver disease, or MASLD, like we call it, and associated fibrosis. And I have organized this presentation into 3 sections. The first being why identifying hidden fibrosis cannot wait. What are the proposed pathways for detecting this hidden fibrosis? And the notes from the field. I hope to share some real-world insights on MASL care pathway implementations, essentially my journey in finding hidden fibrosis in the community. So we'll start with why identifying hidden fibrosis cannot wait. Now, MASL, still often referred to as fatty liver, is fundamentally a cardiometabolic disease. We can define it as the presence of fat in the liver, and clinically with the presence of one of these cardiometabolic criteria. So in our work with the Framingham Heart Study, we found exactly what you are probably thinking. The more the cardiometabolic criteria, the higher the risk that the person has MASL. So where are we going to find these patients? And these patients exist in our primary care clinics, where 40% of patients today in America have obesity in a primary care clinic. About 15 to 20% have type 2 diabetes. This is amongst adults. And about 40% have hypertension, and similarly for dyslipidemia. So that's where I'll find those patients. By the time they come to my clinic in hepatology, there's a very high chance that they have something going on. But hidden fibrosis stays in primary care and has to be found there, in my opinion. So what is MASH, or metabolic dysfunction associated steatohepatitis? It is MASLed with the presence of inflammation, and we'll talk a little bit about that too. So what percentage of patients with MASLed have MASH? And to explore that, I'll go through the natural history of MASLed. And MASL kind of begins with fat in the liver, and it's estimated that 25 to 30% of American population has fat in the liver. 7 to 30% will kind of develop hepatocellular injury, which is MASH or steatohepatitis. And this is the key driver of fibrosis in the liver, which we can quantify histologically, where F0 is no fibrosis, and F4, through progressive stages, F4 is what is cirrhosis. And 4 to 14% of these patients with MASLed will develop progressive fibrosis and cirrhosis, and some of them will even get hepatocellular carcinoma. I show you these statistics, everybody here knows the natural progression because the scope of it is wild, that there are so many people out there and somehow we don't see them. The MASH trials are very hard to recruit. They cost hundreds of millions of dollars and take years to recruit. And when you find a patient that you think has MASL, out of every 4 patients you screen, 3 of them will screen fail for various reasons, but they're very hard to recruit. And you would think that they're all out there. So the drug target, if you have a drug, you want to halt steatohepatitis, and you probably want to reverse fibrosis. And the place where you want to reverse fibrosis is F2, F3 fibrosis in non-cirrhotic patients, that is. And that is where, you have the most tangible benefits. You can probably have a decrease in liver-related events and even decrease in cardiovascular events, and you may even live longer. So those are the patients that I want to find. It's a public health problem, and this slide just shows you the magnitude of it. If we do not treat MASH right now, by 2050, we'll have 122 million people with MASLed, and 23 million of them will have MASH. And this would mean 2 times the amount of liver cancer, 4 times the number of transplants, and liver-related mortality itself will rise in America. And this not only shows the public health importance of this problem, but I can also underscore it by the MASH market. Depending upon whom you ask, it's a $7.2 to $17.6 billion market, and this involves not just therapeutics, but how to find these patients and how to take care of them. There are imaging, diagnostics, antifibrotics. So lots of things come in this market. So it's not all doom and gloom. And I'm happy to report that in March 2024, resmetirom, which is a thyroid hormone receptor beta agonist in the liver, became the first FDA-approved therapy for MASH via the accelerated approval pathway. It's really fitting that 30 years ago and 20 years ago, Rebecca Taub stood here and probably talked to a similar audience in Arias Symposium about thyroid beta receptor globulin. And here I am talking about this. It met the two endpoints that I talked to you about, which is resolution of steatohepatitis and improvement of fibrosis in a meaningful way. But patient needed like 2 biopsies 52 weeks apart. What we don't know right now is that does this drug make patients feel better, function better, and live longer? Those are the FDA endpoints for final approval, right? So the trial is still ongoing, and we'll follow these patients for 5 years. And there's another drug, and it's semaglutide. And in August 2025, this became the second FDA-approved agent, and it also met those endpoints in a meaningful way. And there were 2 biopsies, 72 weeks apart. While we do all these biopsies to see whether these drugs work, I in my clinical practice don't do biopsies. I have to find them, in a non-biopsy fashion, and we'll talk a little bit about that. So this is the conclusion of my first part. Like, identifying hidden fibrosis cannot wait because there's this rising prevalence of MASH. And this fibrosis itself, this F2, F3 fibrosis, is an important predictor of adverse outcomes, adverse liver-related outcomes, adverse cardiovascular outcomes, and death. And the treatment options are increasing, and maybe we can reverse, not just reverse steatohepatitis and fibrosis, but reverse the course of disease and halt or prevent progression to these adverse outcomes. So what are the proposed pathways of MASLD and MASH care? Now, pathways of care are determined by various associations through groups of experts who probably deliberate for anywhere between 6 to 18 months to come up with a paper of what should be a proposed pathway for MASH care. So I think most guidelines now, so we have the endocrinology guidelines and we have the ASLD guidelines. And now, very recently, we also have the American Diabetes Associated guidelines. And they recommend a proactive tiered strategy to recognize and identify patients with significant fibrosis. This is all those above F2 stage. But there are no biopsies here. We have to use non-invasive tests. And who should be screening for them? Everybody who sees these patients. These are primary care doctors, endocrinologists, weight management, and gastroenterology specialists. We all should be screening for significant fibrosis. In home, in the high-risk patients, earlier we used to just look at patients with elevated LFTs and fat in the liver. But we know that we should be looking for fibrosis in patients with diabetes and pre-diabetes, obesity, and particularly if you have 2 or more of those cardiometabolic risk factors. And how should we do this? And that's what I'll focus over the next 2-3 slides. So the primary risk assessment is thought to be simple, easy, a blood-based biomarker called FIB4. And the secondary assessment by any of these 2 tests, this is vibration-controlled transient elastography. This is FibroScan, which you heard a little bit about in the Fontan lecture, or proprietary test called enhanced fibrosis test, enhanced liver fibrosis test. But I'll explain to you what FIB4 and VCTE are. So FIB4 is deceptively simple. It's a non-invasive test for risk stratification. It includes age, AST, platelet count, and ALT. I think the guidelines assume that everybody would have these, right? These are very simple tests, very routinely performed. They're inexpensive, and you could easily make a calculator that you can incorporate into the electronic health record. And it has a low cutoff and a high cutoff. So if you have the low cutoff, you have a very low probability of significant fibrosis. And high cutoff, very high probability. And there's an indeterminate gray zone for which you progress to the secondary risk assessment. FIB4 is a great test. It's not perfect, and most tests are not perfect. Like an example is that, if your age is high, your FIB4 will always be high, right? So amongst other, it doesn't work in a very acute setting. If your LFTs, your AST and ALT are like in 500s, it's not valid. But it's a good test. It's easy. Then you go ahead and do the second test, which is vibration-controlled transient elastography or FibroScan. It's ultrasound-based. It has a little wand, if you see the picture on the side. It sends sound waves into the liver and measures the speed at which it travels inside the liver. It can be used as point-of-care test, and it measures liver stiffness, which here we're using a surrogate for fibrosis. It has its flaws too, as we all do. And one of its flaws is that, like, let's say your liver is boggy. Like if you have a Fontan-associated liver disease, it's going to interpret something else rather than liver stiffness. So 2 cutoffs, less than 8, very low probability of significant fibrosis, and above 12, high. And you come down to that indeterminate, which decreases in size. And, you know, all those patients, you should probably refer to a hepatologist who may or may not do a liver biopsy. So you know the tests, and so this is the screening strategy. So you take the high-risk patients, make sure they don't have other liver diseases like hepatitis C, do the FIB4. If it's low risk, you don't have to send them to a hepatologist. If it's really high, they should get specialist care. If it's indeterminate, get the FibroScan or the VCTE. If it's low, again, don't worry about it. If it's high, refer to hepatology. And if it's indeterminate, also refer to hepatology for further testing. So very simple, you know, very easy to implement, you know, as compared to all the stuff that you showed me. This must be the easiest thing you saw today, I would think, you know. All right. So notes from the field. Okay, so I was like, we're going to implement this pathway. All right. This is what all the guidelines have been saying for the last 3 years. Okay. So, you know, everybody talks about the electronic health record, right? So we made this clinical decision support system, and we're like, we have to do this in a weight management clinic. You know, everybody is high risk for fatty liver. Everybody has fat in the liver. We just have to find the fibrosis, right? Okay, very few people used it. That's the skinny on it. And I'll show you, it took 2 years, okay, 2 years for me to do this. So we made this FIB4-based decision support system, which is embedded in the electronic health record. It had like nice pastel colors, and it said that, hey, your patient has like a high FIB4, click here to get this test, okay? And you didn't have to do much, you just had to click. And there were 14 providers, doctors and nurse practitioners, and we did this educational outreach. All of them attended at least 2 out of 3 interactive sessions where we talked about fatty liver, its burden, you know, why we need to recognize it. And I was around for internal facilitation. You know, if something goes wrong, I would troubleshoot it in real time. And this is our data, okay? The 6 months pre-implementation is in blue, and 6 months post-implementation is in purple. There were like 4,000, above 4,000 patients in the post-implementation phase. The first thing is that FIB4, what I learned is that FIB4 is not really available in everybody. You know, you probably will get it in 50% patients at any given point of time. And we did find advanced fibrosis through high FIB4, right? But nobody clicked, like 9 people clicked on that, please refer to VCTE out of 234. And it's like, we talked about this, like, what happened, right? And then the implementation way to do it is to do a qualitative study, okay? Like you interview them, you code it, you put it in like en vivo. So this is what they told me. They're like, okay, in spite of all your talks, we kind of really don't trust your fibrosis scores, okay, how much ever the guidelines say. And, you know, there's some technical errors in this electronic deployment. We didn't like that, so we're not going to touch that. Okay. And you kind of placed it in this location in the EHR that we don't care about much. You know, there are other things that we do in this clinic. You know, this is not our priority, and maybe the primary care provider should do it. And this was like, this was very important to me, and a revelation that, you know, it really cannot, what it really taught me is that I cannot go somewhere and say that you're going to do this, okay? It really cannot be top down. It has to come bottom up. And everybody who talks about like all these decision support systems, best practice alert, I have a very nice example for them in this. So I decided to like change the way I look at it and, you know, have a different approach, have a single clinician, have a clear ownership of this pathway. And I call it the East Boston experiment. So the FibroScan or the VCT is not really available in primary care centers. And so BMC, Boston Medical Center, has 14 affiliated community health centers. And whenever somebody needs a FibroScan, they actually come to the main hospital. And I saw that this gentleman, Dr. Trillianos, would send about 50 patients in 6 months. And I would be like, he's the only primary care provider who knows what this is, right? So I wrote to him and I'm like, shall we like bring FibroScan to you? Do you want to do this experiment to see whether you can find fibrosis in your clinic? And he said, yes, we're going to do it. So we wrote a grant and we got some money and we got the FibroScan, which is the important one. And I call it the East Boston experiment. This was done in Neighbor Health. This is a federally qualified healthcare center in East Boston. It takes care of about 56,000 patients, adult patients every year. Most of them are socially disadvantaged. Most patients are Hispanic, 85% are Hispanic, maybe 50% are below the poverty line. They have very high burden of cardiometabolic disease, particularly obesity and diabetes. So it was a 4-strategy bundle. We had a clinical decision support system which primary care providers could look at and understand who is their patient. We did provider education. Dr. Trillianos would do the VCTE. We were there with him. We taught him how to do it. We made sure that the testing is linked in the electronic health record system. If there is a test, the patient knows the results. And if there are results are positive, they are linked to care. And we were there to address workflow issues, provide updates, and offer support in all technical difficulties. And, it seems like a nice strategy. It took 6 months of partner alignment. This included evaluating operating requirements. It included fiscal and business analysis, like crash course for me, defining what the IT infrastructure would look like, and what integrations are needed to support this pathway. Just none of this I was trained for ever. But yeah, so that was mostly what I did. And this worked, this worked. The number of FibroScans in 6 months were like 308, which was a 431% increase. 21% had high liver stiffness. And it didn't matter whether you had liver stiffness, because even if your liver stiffness was low, 26% actually had a change in management of their care. Like, they were referred to bariatric surgery, they went to weight management clinics, they got GLP-1s. Some got vitamin E, and 13 patients were actually enrolled in my clinical trials. So this was 6 months. I would call this a success because East Boston told me that they're getting their own machine, and thank you very much. We can do this on our own. So that I believed was success, and I thought that that should happen at Boston Medical Center. Why can't I do it at BMC? I can do it in in a community health center. So we thought of a different approach. There was very little interest in primary care doctors to do this. So we tried this task shifting of MASLD risk stratification to pharmacists. Now, diabetes care has ambulatory pharmacists who help providers, primary care providers, in taking care of their patients. They titrate medications, they take care of routine screenings, and they are the ones who would do this fibrosis risk stratification. We did pharmacist education. We brought FibroScan to their clinic. We got a portable machine. And we also helped with internal facilitation. And 6 months down, this was also successful. This shows that in the pre-intervention 6 months, there were no referrals to FibroScan. Then no risk stratification was done in any patient with diabetes. But in this small clinic, where pharmacists saw 224 patients in 6 months, FIB4 was available in 61% patients and was elevated in 45% of them, very high in diabetes. And 45 FibroScans were completed, and we did find 8 patients with advanced fibrosis who received treatment for fibrosis, and 6 of them actually had cirrhosis of liver. So it's hidden. If you look for it, you're going to find it. And as I think of, advancing, to develop type 2 hybrid trials that I can implement across community health centers and write a grant for it, and do system-wide implementation in BMC hospitals, I have a list of lessons learned, which I incorporate as I write my aims typically. It takes a lot of time and sustained effort, and it is really important to engage leadership early, and you require institutional buy-in. FIB4 and VCT are imperfect, but they are a very valuable starting point, and we cannot wait for that perfect tool to find these patients. Workflows really should come from the people, and they should be for the people and by the people. And providers really value this end-to-end solution. So if you find the patient, you should do something with them and minimize the burden on primary care doctors who are very overwhelmed these days. So just telling them to do this pathway really will never work, I think. And I had this final thought. I work with implementation scientists, and you guys have this final endpoint, right? Like, this is your drug target, this is your mouse model. Like, they have this model, okay? Like, the goal of every implementation scientist is to have this framework which they can implement, theories, models, and frameworks. And they're probably going to roll up their eyes to look at this, but have you heard of Ikigai? I saw this book at an airport, and it talked about like what brings joy to your life, right? So what brings joy is apparently like, this in this framework, you have to do what you love, okay? You have to do what you are good at, and the world should need it, and you should get paid for it. Okay. All right. So there is this, my 2-year journey in Maslow risk stratification, I think there's this ikigai for Maslow risk stratification. You have to be a believer. Okay? You really have to be a believer. And then you have to be ready. All these people, all these partners and stakeholders should be on the same page. You have to be ready. It's really important. You can't like rush this. And you have to have a referral plan. You can't find all these patients and not do anything with them. And you do have to get paid for it. Okay, so as I design strategies, I think of how this will benefit primary care clinics. And I hope my type 2 hybrid trial can show that. There are all these people who help this happen. And I thank my patients who make it all this worthwhile. Thank you very much.
From the study that you did in a primary care, would you predict that many patients would be diagnosed with significant liver fibrosis and disease? Is that predictable?
So I think the biggest bang for the buck is in people who have pre-diabetes or type 2 diabetes, because insulin resistance is so central to the pathophysiology of Maslow. Then I would find cirrhosis in those. I mean, this is not once. I would find cirrhosis that I would tell them for the first time in primary care. Let's say about 20% amongst patients with diabetes.
That was really cool work. Thank you. I think it highlights to us, we've been talking about in its different names, fatty liver, for 30 years. And if you have a solution that we now have with therapy and we have diagnostic workup, but we tell both the primary care providers and the patients once that actually you still have to wait 8 months to see a hepatologist, everyone must feel like it can't be that bad and that urgent. And I think for you to put that urgency into the clinical practice that I think we all feel in the lab, in our mouse models and in our studies is really amazing. Congratulations.
Thank you so much. Thank you.
If there are no more questions, we'll move on to our last 2 Microtalks.
Thank you.
Our first Microtalk is from Shijun Ding and the Hotem Cytological Lab at Harvard University.
Thank you for the opportunity to present our work here. It's a great honor. We are interested in the role of cholesterol in the MASH progression. Emerging evidence suggested that cholesterol overload is one of the major contributing factors for MASH progress from simple steatosis to liver inflammation and fibrosis. We have identified a ER-resident protein called NFE12. It directly binds to free cholesterol at the ER membrane. When we delete it in the hepatocyte, it causes massive cholesterol accumulate in the liver, and together with liver inflammation and fibrosis. Here's what the liver looks like in the knockout. And mechanistically, we found it in response to free cholesterol rising. NFE12 interact with the SRBP gatekeeper INSIG1. It promotes the INSIG1 degradation and liberate SRBP activity for cholesterol secretion. And when we remove one of the small motif at the N-terminal domain of the NF12, this degradation is blunted, and the NHB2 domain have the similar function of the wild type. And to test the in vivo physiologic function of this interaction, we put this wild type or mutant protein back to the knockout liver and perform cholesterol secretion assay. And the wild type protein fully restored the cholesterol secretion function, and the mutant lost this function. It tells us this interaction not only important for cholesterol secretion, is this binding motif is very critical. Then we want to test it in a disease model. We overexpression NF12 in a MASH model, and we found that overexpression reduced the cholesterol content in the liver. and more importantly, reduce the marker for liver injury. It together with the reduce liver inflammation and fibrosis. In summary, our finding suggested that the NFP12-1 interaction may act as a cholesterol response in a checkpoint to maintain cholesterol homeostasis. Moderate this action may be another potential strategy for MASH. Yeah. Thank you.
Thank you so much. Our last Microtalk speaker is from Martin Furhard at the Lauer Lab.
Thank you for the introduction. I would like to talk about hepatitis B. And since we haven't talked a lot about viral hepatitis so far, I feel a little bit like the odd one out. Let me tell you, hepatitis B is caused by contraction of HBV, and it has a dichotomous outcome. There is acute hepatitis B, which you mainly find in adults who contract HBV, and they can control in 6 months, they can control the infection. Control here means that the body, the immune system, is able to control viremia and antigenemia, but there's still DNA in the nucleus of the hepatocytes. And secondary, there's chronic hepatitis B, which you mainly find if children or infants are infected or immunosuppressed people. And people with chronic hepatitis B have an increased risk of cirrhosis and HCC. Chronic hepatitis B can also be controlled by the immune system, but it takes 4 to 6 decades to do so. So there's a big difference. And as far as we know, the dichotomous outcome is caused by immune cells, which is why we want to address this gap in knowledge and look at this. The main problem is that most people study the immune system in the blood, but we know that the immune system in the blood, or the immune cells in the blood, and the immune system in the organ that is actually infected in the liver are different. That is why we have a big study in Zambia, where we're looking at 233 patients with acute or chronic hepatitis B so far, and we have longitudinal samples, blood draws every 3 months, and liver FNAs every year. Starting at the day of treatment for chronic hepatitis B or acute hepatitis B at the day of enrollment. And this is how it looks like. So we're doing the liver FNA in Zambia, capture the cells and load them into these hives so that we ship to Boston, and then do the library preparation and single-cell transcriptomics here. And at the moment, we have 190,000 cells in the object. Here you can see a UMAP depiction of this with the different cell types that we capture, and you easily see that we have very little non-immune cells. This is because hepatocytes can barely be captured from FNAs. And in the stacked bars, you see differences between acute hepatitis B and resolved or controlled hepatitis B. And you see that upon control, we have less CD8 T cells, but more NK cells and more granulocytes. And additionally, in acute hepatitis B, we see T cell activation and more ISG signaling in the immune cells, but less ISG signaling in hepatocytes. While after control of acute hepatitis B, we have more cytotoxic T cells. So we think this is mainly dependent on coenzyme B, 6,3-CR1, and less ISG signaling in the immune cells again. Now we're also looking at how HIV co-infection influences this, because we have a subcohort for that. Thank you.
Thank you to all the speakers for this session.
Our first speaker today is Dr. Vadim Gladyshev. He is a professor of medicine at Harvard Medical School. He's the director of the Center for Redox Medicine at Brigham and Women's Hospital, and he's also a faculty member at this institute. His lab has focused on the study of aging, rejuvenation, and lifespan control using a combination of both experimental and computational approaches, and that work has resulted in the publication of approximately 500 articles, and Dr. Gladishev's receipt of a variety of notable awards, such as the NIH Pioneer, Transformative, and Eureka Awards, as well as his election to the National Academy of Sciences. So without further ado, I'll welcome Dr. Gladyshev to the stage.
Thank you very much. Thank you for the kind invitation to speak to you today. I will tell you a little bit about our studies on aging, longevity, and rejuvenation using liver as a model system. A couple of years ago, I chaired a Gordon conference called Systems Aging. And this is a conference on aging. And we gave a questionnaire to speakers and attendees and asking the most basic questions, like, what is aging? What causes aging? When does aging begin? Is aging a disease? And questions like that you see on the left. And this is like a word logo, how people answered. Basically, there was no consensus on any question. And when I try asking on definition of aging, there was 106 responses, there was 106 definitions. And I try to cluster them. And so some people think of aging as increased chance of dying. Other people say, no, it's like loss of function. Or maybe some people think development is a period after completion of development. Other people say changes in biological age, or maybe accumulation of damage. Basically, there's no consensus. It's a problem. Sometimes we say, what do you study? Aging. I study aging, but maybe we study completely different things. So one way to think about this process is that we could study aging in terms of mortality, but we know that mortality is caused typically by diseases, and then something like upstream is functional decline, and ultimately this decline is caused by damage. So my thinking is more on the damage side, on the molecular side, but eventually kind of leads to all of these features. For example, dying maybe not just because of aging, but for other reasons as well. And so it's very important, I think, in the aging field at least, to have like a kind of a concept, something like a model in mind before we do experiments. With this, we organized our program, research program in the lab. So we study longevity, which is something shown on the left. So on the left here, longevity, for example, longevity across cell types. There are some cells in the body, they are very short-lived, like cells in the blood, and some are very long-lived, like neurons or cardiomyocytes. And of course, they have same genome, same origin, but then widely different lifespan. We can study differences and also longevity across mammals. Some mammals are short-lived, like shrews, they live only 1 year. Some are long-lived, like some whales, they live 200 years. They have the same ancestor, but something changed in their genomes. Now they have widely different lifespans. Nature is able to change lifespan very easily over evolutionary timescales, and we study that. Or longevity across interventions, because we now know many ways to alter lifespan in model organisms, not in humans, but in model organisms, and we can study that. This results in what we call signatures of longevity, which are omics patterns, which are indicative of the potential of the system to live long or short. These are not age-related changes. This is the changes that are associated with change in longevity, potential to live long. But we also study aging here on the right, and here are the age-related changes, and we need to quantify them. So for this, we developed tools which are called biomarkers of aging or aging clocks, for example, epigenetic clocks or transcriptomic clocks or organ-specific clocks. I will tell you a little bit about that. And the third component is rejuvenation here. Rejuvenation is transition from an older state to a young state. Because we have to realize that aging is a very complex process. It's probably irreversible because there are so many changes there, like, for example, mutations. You cannot reverse mutations, but not just mutations. But nevertheless, we could target a system so that it transitions from an older state to a younger state. We call it rejuvenation. And with these three components, we are able to target aging and rejuvenation, identify like dietary, pharmacological, genetic, lifestyle changes that are associated with changes in the rate that we age, or maybe in the biological age of the system. And I will give you a few examples of that. First is this example from a paper published a couple of years ago. This is longevity across mammals. We analyze 41 species of mammals, multiple representatives and multiple organs through omics approaches. And in this particular figure is based on the transcriptome. So it's a large RNA-seq kind of project. And you can see these are mammals, they differ in life history traits, including lifespan, about 6, maybe to 8, 10-fold. And this is changes in the liver. We identify many genes, expression of which correlates with lifespan. You can see, for example, this gene, like RPL28, increases with increase in longevity in the liver, and this one increases, and this is decreases. What is interesting that when we find these genes in the liver, we look for them in the brain or in the kidney, they go in the same direction. I think it's counterintuitive, but for some reason, if a gene changes in one organ in one direction with longevity, it typically changes in the same direction in other organs, even though the biology of the organs is very different. Another example is where we analyze this across interventions. This is the data, the transcriptome in the liver, and this is the interventions that are known to extend lifespan. Some of them are genetic, like Ames mice, little snail mice, growth hormone receptor knockout, FGF21 overexpression, or calorie restriction, methionine restriction, every other day feeding, and so on. And this is Spearman correlation matrix. So you can see that immediately there's a cluster that means that these interventions, dietary or genetic or pharmacological, they change the system in the same direction. They kind of change similarly the animals, and they become longer lived. But there are some other interventions which change the system very differently based on the transcriptome, yet they also result in extension of lifespan. For example, rapamycin here, you see, it doesn't correlate with anything. This one is more like calorie restriction cluster, but there are other ways like rapamycin or like CMYC deficiency and so on. And now we have these changes across interventions, changes across longevity of mammals, and we add a third component, aging component, which is transition from younger to older state. Let's now compare these three components. Yeah, and this is shown here. This is longevity across mammals, like brain, kidney, liver, and residuals of these functions. They cluster with each other. These are aging changes like muscle across mammals, brain across mammals. Human means different tissues of human. Global means different species and tissues. They also have a cluster. Yeah? And this is intervention cluster, like various longevity interventions. But now we compare these three clusters, and quite interesting observation that emerged from this. First, aging changes are negatively correlated, blue is negative, red is positive, with interventions, longevity across intervention, interventions that result in longevity. It's reasonable because if we subject animals, for example, to calorie restriction, they age slowly, and therefore they are younger at a certain kind of chronological age than controls. They are negatively correlated. That's reasonable. However, when we compare longevity across mammals with aging, there's a positive correlation. It seems like longer-lived animals are older. This makes no sense. Why would be like longer-lived animals older than like short-lived animals? We think they're not older, but they are better protected. For example, during aging, damage accumulates, but organism responds to this damage, activates protective responses, and these same protective responses are used also in long-lived species. They are just better protected. What is also interesting when we compare longevity across mammals with longevity across interventions, and there is no correlation. This is really surprising. So because we think that we could study animals as model to study longevity, but mechanistically they are widely different, they're completely different. So somehow what we find in the lab, simple interventions like genetic manipulations or dietary or pharmacological, is completely different from how nature extends lifespan. Nature presumably changes many genes and pathways and systems and organs, and this is just not recapitulated in the lab. So to me, it represents an untapped potential of studies, cross-species studies to understand longevity. And maybe in the future, if we understand how exactly nature does it, maybe we could utilize it in model organisms in new ways. I'd like to briefly mention that we could use these longevity signatures to identify new interventions that extend lifespan. For example, this study, which is still under review, we started with in silico screen and followed a screen in cell culture. Then like 25 compounds actually made diets and gave to mice for a month and followed by omics, and identified 10 compounds this way that we predicted to extend lifespan, tested them for lifespan, and 5 of them extended lifespan. So it seems like that system might be used to identify new interventions. It's just one example here, some kind of KU compound. So we predicted it extend lifespan and checked it in old mice. It does extend lifespan. It also changes frailty, kind of improves frailty in old mice. Now I'd like to tell you a little bit about biomarkers, because it's a true revolution in the field. They're called biomarkers of aging or aging clocks. It started with Steve Horvath, used to be at UCLA, now he's at Altos Labs, published a seminal paper in 2013 on the first human epigenetic clock that predicts biological age. But now this field has grown, and clocks, I mean, I'm sure everybody has seen clocks. It's just published every day, many clocks, which is impossible to follow. It's just, and everybody claims that their clock is the best. So to kind of rationalize the field, we formed this biomarker of aging consortium, and it's a large group of people now. This is our first paper, kind of conceptual paper published, but since then there are many papers published. We have a conference, we host a conference at Harvard Medical School. This is just last month, 400 people from 30 countries, and it's just it's a platform to discussing what is biomarker of aging, how we could train them and utilize. And I'll give you a few examples of how we do it. First example is here. This is the work of Jesse Paganik. He found that biological age not just steadily increases with age, with chronological age, but fluctuates. And particularly, it responds to severe stress. Under conditions of severe stress, it increases rapidly. And once the stress is relieved, it decreased. And in this paper, we described 4 major cases, like major surgery, severe COVID-19, pregnancy, and parabiosis. For example, in pregnancy, we find that women in the 3rd trimester, they are biologically older, they increase biological age. At postpartum, the biological age decreases back to approximately normal level. And it nicely corresponds to like gestational diabetes, like heart disease and other clinical phenotypes. So one issue with the clocks is causality. This is actually still unresolved issue because during aging, kind of everything changes. And we could use any of the features to quantify biological age, whether it's omics or even like visually, you see people, you can guess the biological age. However, there are many changes which are, we could use them to quantify, but we don't know if they are causal. Yeah? So for example, if we could quantify changes like number of visits to doctor, wearing glasses, hearing aid, and so on, we could quantify biological age, but it does not represent biological age, right? So it's just other features. And so Albertine, he developed recently the first causal clocks. It's a causality-enriched clocks. And the way he did it, he developed genetic approaches, combining Mendelian randomization with age-related changes. And he developed clocks, like 2 types of clocks. One is based on damage that increases with age. We call it damage clock, or for damage. And this one can be used to quantify biological age in human, this actual age, predicted age, and adapt age. These are the changes that are response to damage, adaptive changes. And it can equally predict biological age. But the difference is that when we apply them in the case of some manipulations or some kind of interventions, and one example is shown here. Here we take cells and convert them to iPS cells, from somatic cells like old cells to young cells. And damaging clock, it shows rejuvenation, but adaptive clock does not. So which is exactly how we predicted. And it just tells that even though some biomarkers, they can quantify biological age, they are less useful when we test interventions because we don't know which component of the clock is affected. Now, I'd like to mention, this was about epigenetic clocks. Now we also developed schistomic clocks, still unpublished also. The Alex Shishkovsky, here he collected over 4,000 samples and of cases where lifespan is decreased or increased, all kinds of different interventions across species. And he developed a clock that predicts biological age based on RNA-seq profiles. We are interested in this because like RNA-seq is, everybody does it, and it can be used actually to predict biological age. The actual age, predicted age, you can see training and test set, very good correspondence. And he also trained the clock not just for chronological age, but for time to death. Yeah, we call it mortality clocks. And this is mortality clock, also training and test set. And this is one application of the clock where we test them with lifespan shortening interventions and lifespan extending interventions. You can see the clock can distinguish them. So in the future, it can be used to test new interventions for the potential effect on lifespan. Also, when we apply these clocks to disease models in mice, we find very interesting, like regardless of each panel has showed different disease, age-related disease, and in model systems. Control disease, control disease, control disease, control disease. In each case, disease tissue is older. It's very interesting. So whatever is age-related disease, we test with control, it's always older. Okay, I will come back to this point late in my talk. So I briefly wanted to mention a couple of tools that we developed so that can be used by the community to test biological ages in different situations. Like one system is called BioLearn, just published last week in Nature Aging. This is a system for harmonization of clocks and data sets, because currently it's just the batch effects and correspondences between the labs is just too different. So this is a system that can be used to quantify biological age. Another system is clock-based, where we basically apply different types of clocks to major databases. And the third one is called MethylGPT, which is a foundation model for denamethylation that can be used also to estimate biological age. So I will tell you a little bit about this clock base. This is the way we did it is through massive AI agent mining. So we developed this multi-agent systems. When there's one agent, for example, reviews literature, another one proposes hypothesis, tests the hypothesis, and up to the right in the paper. And so we applied this to GEO database, 2.5 million samples when we applied different clocks, drugs and diseases and genetic changes, environmental changes, you can see here. And this is an application in terms of biological age. This is, see the middle is here, 0. And so most of the changes we observe, regardless of whether genetic, drug, or environmental diseases, increase biological age. If we manipulate a system, typically it becomes older because it's easier to disrupt. But sometimes when we change the system, it becomes younger here as well. So it allows us to identify new candidates, basically. And this is also an illustration of how we do it. We identify changes that increase biological age, and here changes that potentially candidates for decreasing biological age, and all kinds of different drugs and diets and so on. And we wanted to test some of them. We chose just one of the top candidates. There was nothing known about this in terms of agent called obain as a chemical compound. This is a predicted biological age in young mice. This is in old mice based on the liver data, the top data. And when this drug is applied, actually biological age predicted to be lower. So it was very interesting. So we applied this to mice. And this is a control mouse, old mouse. When we treated with obain, it just visually looks better, like based on the hair. We tested frailty. Frailty is decreased when we treat. We also took 20-month-old mice, treated them for 3 months, up to 23 months. In control mice, untreated frailty increases because the mice are old. But in the treated mice, there was no change. So apparently, this compound is a really good candidate for anti-aging effect found through this kind of mining of the entire GEO database. And if you have any interest in compounds, you can just go to this system that we developed and check them, because the entire GEO database has been analyzed with clocks, and you can simply visualize and make figures for your papers. So now I'd like to tell you a little bit about rejuvenation. So far we've been discussing aging. This is illustrated by this water flowing in the vertical direction is aging. We can slow it down by calorie restriction, rapamycin, whatever, metformin. But what we also want to do, we can take this water and go like upstream, make an organism younger. Yeah? And so we already know one example is iPSCs. So of course the Yamanaka's discovery is conversion of somatic cells to embryonic cells. But for us, aging biologists, it's conversion of old cells to young cells. Because embryonic cells are young by definition, it's rejuvenation is possible. This already tells that rejuvenation is possible. I will give you 2 examples, additional examples of rejuvenation. One is we use this method called heterochronic parabiosis. When we connect mice, like young mouse and old mice, connect them so that they develop joint circulation and keep them for 3 months. And this is done by Bohan Zeng. And what we find is that this is, again, so based on the liver, and this one is the epigenetic age, based on the DNA methylation patterns. But the same, transcriptomic shows the same. Basically, this is when we connect young to young mice and young to old. We see there is difference. But when we connect old to young for 3 months, we see that this liver becomes biologically younger compared to the old liver. And then we disconnect them and keep them for 2 months more. And we find that the effect is preserved, although diminished a little bit. The liver is still younger 2 months later after we separate from the young mouse. So we also tested various times of treatment through parabiosis. So when we connect them for 5 weeks, we see a significant rejuvenation effect, but it's much weaker than when we connect them for 3 months. So apparently, short-term parabiosis is less efficient than the long-term parabiosis. And now we are doing experiments in the lab when we connect them through multiple rounds of parabiosis. Like we connect them for 3 months, give them a rest like 2 months, and then connect them for another 3 months. But this one is still in progress. We don't know. Hopefully they will be super rejuvenated, but we don't know yet. And we also found that this approach allows to extend lifespan. So old mice, after they are connected to the young for 3 months, we separate them and keep them until they die. They are longer lived as well. And I'd like to give you another example of rejuvenation. This is something that we also discovered recently. We call it embryonic rejuvenation. And again, this is applying the clocks, aging clocks. We found that at the beginning of life, at the stage of zygote, which is the fertilized egg, biological age is low, but not 0. It's slightly above 0. And then initially during early development, it decreases to the point we call it ground zero, that corresponds to the lowest biological age of the organism. And we think aging begins from this point. Like in humans, it's about 3 weeks of life, corresponds to this embryonic stage of gastrulation. And our clocks, whether we apply bulk epigenetic clocks, or we also developed single cell epigenetic clocks, also shows the same, and also trichotomic clock, they also show an increased biological age. So mechanistically, we don't understand how exactly this rejuvenation happens, but we are very interested in this because we hope that if we find maybe in the future we could apply it to somatic cells, like adult cells, if we understand mechanistically this process. And we published a few papers I'm not going to discuss, but basically phenomenon is there, but mechanistically there's no understanding yet. Finally, I'd like to tell you a little bit about proteomics, how we use proteomics to assess biological age. And this is the work of Ludger Gemini, who is a postdoc in the lab. He used this UK biobank data set to train a clock based on the plasma proteome. And this is the men and women shown in different colors, the actual age, predicted age. You can see there is very good correspondence. This is a clock trained for chronological age, and this is a clock trained for time to death. Also works really well. But then we thought that instead of talking about biological age of the organism, maybe we can talk about biological age of different parts of the organism. And we wanted to develop these organ-specific clocks. And this is an example from a paper we published early this year. So how do we do it? So first, we use this method that developed by Tony Viscaray from Stanford a couple of years ago, when he basically proposed that proteins in plasma, they come from different organs, right? So if we identify where they come from, for example, we find that 200 proteins in the plasma, they are unique to brain. Yeah? And then follow how they change with age, we could use that to predict biological age of the brain. Yeah? So now when we profile plasma and identify the source of these proteins, of course, not for all proteins, some proteins would come from different organs, they're not very, very useful for us, but some are quite unique. We can tell biological age of different parts of the body based on the plasma proteome. And this way we have clocks, for example, liver clock, brain clock, artery clock, or lung, immune, kidney, intestinal clock, and so on. By profiling plasma proteome, we tell age of the different parts of the body within the same organism. And they correlate to one another. These are residuals of the clocks. This is the chronological model, and this is the mortality model. Mortality actually works better than chronological model often. And of course, they correlate. If somebody is older in the brain, typically that's the same person is older in the liver or in the kidney. That's reasonable. But what is interesting is when we apply these clocks to diseases. So for example, we take this liver-specific model and ask a question: which disease is predicted best? And we find that liver cirrhosis is best predicted by this model. Yeah? Especially for this mortality clock. You can see this hazard ratio is really amazing, more than 2. This is a chronological model, and this is a mortality model. So liver is much better than mortality is predicted. The same way we observe in other organs. So for example, here, we analyze association between diseases, all kinds of age-related diseases, and organ clocks. Yeah? And we find, for example, look at people with dementia. Blue is young, red is old. People with dementia, they have low intestinal age, they are young in the kidney, in the artery, in the skin, everywhere. They are old here, brain. People with dementia, they're old in the brain, but not old in other organs. Or like people with COPD, it's a lung disease, young, old in the lung. You see? So it seems like disease again shows an accelerated age in an organ. So this is our model. So this is zygote. This aging begins here we call it ground zero, 3rd week of life in humans. And then kind of organism develops and ages somewhat synchronously, different organs, yeah? But then sometimes one organ or more deviate from that. They, for some reason, become older. It may be genetic predisposition, some lifestyle changes, or stochastic changes, becomes older, yeah? But it's not simply older, because it's not in isolation, it's part of the organism, so it interacts with other organs. In a way, other organs in a young state, they pull it back, they don't allow it to be old enough. So now this organ, like liver, heart is shown here as an example, is both older and altered. And that condition we call a disease. In this paradigm, there are no diseases. All we have is aging. And it would be interesting to explore this model in the future, of course. So finally, I'd like to summarize what I discussed in my talk today. First, we discussed a little bit about aging. I told you about the beginning of aging, although we don't fully understand the aging process itself as a community. And I also think aging is not adaptive. There is no program for aging. There is no aging genes. It just happens like a default state. I told you a little bit about longevity signatures across species, cells, and interventions, and also differences between species longevity and longevity across interventions. Also, I gave you one example of how this can be used to identify new interventions that can extend lifespan. Also, we discussed aging biomarkers, which is a true advance, like major advance in the field. I mentioned about bulk, single cell, causal, stochastic, and multi-tissue biomarkers, also at the level of epigenetic clocks, transcriptomic clocks, and proteomic clocks, although we also have like imaging clocks, for example, or some other clocks. Also about reversible changes in biological age upon severe stress. I gave you 3 examples of rejuvenation. One is cell reprogramming, for example, Yamanaka factors, and also chemical compounds. We published a couple of papers on chemical compounds as well that allow rejuvenation. Embryonic rejuvenation, which is a very new discovery culminating in ground zero, although mechanistically not understood, and also an example of heterochronic parabiosis. And also I discussed organ-specific aging, organ aging models, chronological immortality, association. I didn't discuss and diets, but I excluded that slide, but anyway, so diseases can be viewed, I think, as pockets of non-autonomous aging. But all we do is actually to try to understand aging, we try to understand and target aging, longevity, and rejuvenation. We are still like in the middle, or maybe even at the beginning of that long road, because it's a difficult problem. Aging is a very difficult problem. It seems very simple on the surface, but once you study, it just becomes too complex. But anyway, so it's very exciting to work in this area. I think we see a huge influx of resources, talent, like opportunities. So it's really an amazing time to be in the field. And this is my lab, current lab. And also I mentioned a few studies, also former lab members. All of our studies are done in collaboration. We have many collaborators and also funding sources. Thank you very much.
Thank you so much. That was so inspirational. I wanted to ask you a question about your last part of the talk, organ-specific aging. And the clinical implication of that came home to me in recent weeks when we had liver donors who were 85 and 87-year-old. Most transplant programs were rejected because they are over 75. But in both those donors, the patients who were transplanted in their mid-50s went home after a week. And so the question is, in your harmonization of biomarkers, do you have any suggestion of perhaps how do we look at liver aging of potential liver donors? And second is, let us say if we have a donor liver, if it was perfused with huabane, can we reverse its aging?
Yeah, it's a very good question. Actually, we are working actively on that in the lab. We use both models, like transplantation models. We have a couple of surgeons in the lab where we transplant different organs. We are still learning about the liver, so how to transplant, but we have data on the heart. So, and we find that when we have a model, it's collaboration with Stefan Tullius, who's a head of surgery at Brigham Women's Hospital. So we have a model of mice with 2 hearts. Like we have like young mouse, one young heart, and we put a second heart like old heart. And looking at the changes in the biological age of the young heart and old heart, we find that old heart is rejuvenated in the mouse. And we also see in human patients, because when the human hearts are transplanted, the biopsies are taken. We analyze these biopsies, we can see that old heart in the young body is rejuvenated. Not fully, of course, it's partially rejuvenated. And I think it's very important because, again, as you mentioned, old organs are typically discarded, but actually they potentially could use because they may be rejuvenated.
Hello, I have a question about, you showed a older mouse connected to a younger mouse in one of the slides. And I was wondering if there was any instances of potential rejection of the additional body, almost like organ transplant, or if, as you mentioned, stress can increase biological age. Did that have any significant impact on some of the mice that you saw?
Yeah, excellent question. First, in mice, we use inbred mice, so black 6 mice, so there's no organ rejection because it's a model system, right? So it's not perfect, but that's the way it's used. But you're right that this procedure itself, whether it's a parabiosis or organ transplantation, is stressful. It actually by itself increases biological age. So there's a slight increase in any of these procedures. Yeah.
One of the mammal that lives like really long, I think, is glass sponges. I think lives like 1,000 years. Can we learn something from those glass sponges, that live forever, and implement some kind of ideas of what we think we could change,
Yeah, I mean, I'm very interested in kind of cross-species analysis. We published a lot of studies on that. I am not very familiar with that particular organism, but there are other long-lived organisms, so some potentially non-aging organisms like some planaria, for example, or hydra, and some other organisms, but they typically don't have like fully differentiated non-renewable cells like mammals have. So mammals presumably would unavoidably age simply because the way we are organized, like we have skeleton, eye, cardiomyocytes, neurons. That's it. We should age. We could slowly age, but we will age. But the other organisms, they can replace any of their organs and parts from stem cells, and they potentially might be in the non-aging state. There are also very extremely long-lived species, very interesting. For example, across mammals, the most interesting one is called naked mole rat. We also study this in the lab. They don't show increased mortality with age, and they age very slowly. It's the size of a mouse, but lives more than 40 years. But it does age based on the epigenetic clocks, just slowly.
Our next speaker of the session is Dr. Sammy Wang, who is an assistant professor of medicine in the Division of Liver Diseases at Sinai in New York. During their postdoctoral training under the co-mentorship of Drs. Kirsten Sadler and Scott Friedman, they discovered an epigenetic crosstalk that governs mammalian liver regeneration and identified an autocrine signaling circuit that emerges in hepatic stellate cells during advanced liver fibrosis. And they have continued that work under their current NIH-funded research program, which focuses on the epigenetic mechanisms that regulate cell identity and memory in the liver. And the ultimate goal of their laboratory, as well as I think the joint hope of everyone attending this meeting, is the translation of fundamental discoveries in liver biology into treatments for patients with chronic liver disease. Without further ado.
Thank you, Alex, and thanks to organizers for inviting me. As a trainee, I've always attended this symposium, so this is quite an honor to be here to present to you today. So the title of my talk is Hepatic Cell Plasticity and Maladaptive Fibrogenic Memory in Chronic Liver Disease. And my name is Sammy Shuang Wang, and I'm from the Division of Liver Diseases at Mount Sinai in New York. So just a very brief overview of chronic liver disease. So as most of you already know, the liver, in response to chronic injury over many, many decades, develops chronic disease. And this injury comes in many forms. We've talked about today fatty liver disease, but also viruses, but also alcohol exposure. And in response to this repeated injury, the liver develops chronic inflammation and also progressive fibrosis. And the liver can exist for many years in the state of chronic liver disease, but ultimately it succumbs to these very severe end-stage diseases such as cirrhosis, liver failure, or liver cancer. What is also very interesting about the liver, and I think this is made very clear by the miracle treatment, the HCV therapy that we have now that completely clears the HCV virus. What is shown very clearly is that even when the injury has been occurring for decades under HCV infection, once you terminate that injury, that liver can actually spontaneously recover. And this is shown both in the resolution of inflammation, but also sometimes a regression of these very thick, dense hepatic scar that has developed. So I think this diagram really sets up the premise or the clinical premise of the investigations that I've undergone in my lab and also part of my postdoc. And these are mechanisms that we continue to explore with the hope of identifying some new treatments for chronic liver disease. So since part of my training took place in Scott's lab, we are very interested in studying fibrosis. And Dr. Scott Freeman has identified several decades ago now that the most fibrogenic cell in the liver are the hepatic stellate cells. So this is diagrammed here. The stellate cells, which are normally quiescent, are actually quite beneficial for the liver, would activate in response to injury. And they activate into these myofibroblast-like cells called activated stellate cells. And these cells produce a lot of ECM, like other myofibroblasts from other tissues. What was traditionally thought is that these hepatic stellate cells, like other myofibroblasts, are very short-lived and they will actually just apoptose after the injury subsides. However, a pair of studies about 10 years ago using lineage tracing in mouse models have very cleverly shown that the activated stellate cell actually don't all apoptose. Roughly half of them deactivate and return to a quiescent-like state. So this observation is very important for the story that I'll tell you about today, because this suggests that these cells are relatively long-lived in the liver, and this gives them the capacity to carry some sort of cell memory. So a lot of the work that I've been doing now really started in my postdoc, because we essentially found out that using single nucleus RNA-seq, we're able to capture gene expression of a lot of stellate cells. So this is quite a dramatic improvement over single cell RNA-seq. So then, since then, we have applied single nucleus RNA-seq to really study and understand the transcriptomic changes that occur in the stellate cells as MASH fibrosis develops or as fibrosis develops. And this led to the postdoc paper that I published in Scott's lab, where we uncover an autocrine signaling loop that takes place among stellate cells, that they undergo the switch from paracrine signals, signaling among other liver cells in the liver, to a very independent of other cell types, and they engage in this mainly autocrine signaling modality, and this occurs in late-stage liver disease. And once we identify this, we are able to subsequently show that if you perturb the autocrine signaling of stellate cells in late-stage disease, you can actually reverse very advanced fibrosis. So that was part of my postdoc work. We then applied this similar technology to the right side of the diagram. So we wanted to ask, can we apply single nucleus RNA-seq to understand the cellular and molecular drivers that actually underlie fibrosis resolution? So as I mentioned, if you terminate the injury, the liver can spontaneously resolve the fibrosis and also the underlying disease. But it is unclear what are the mechanisms that drive this process. And specifically, what are the processes that actually turn over the matrix that make up the hepatic scar? So in this study that is already online as a preprint, and hopefully you will see published very soon, we've essentially identified a subpopulation of endothelial cells, which we call scar-associated endothelial cells, because we found that they penetrate very deep into the scar, and they're actually vascularized within the scar. And these so-called scar-associated endothelial cells not only crosstalk very extensively with the stellate cells through Wnt, Wnt9b to be exact, but they also actually demonstrate direct protease activity, and this is work in collaboration with Sangeeta's lab. So we think these scar-associated endothelial cells, which have been relatively overlooked until then, are actually a very critical drug target for perhaps promoting fibrosis regression in these patients that don't have or unable to spontaneously resolve their fibrosis. So in subsequent studies in the lab, we're going to follow up with that study and hopefully identify specific delivery mechanism that can actually target these scar-associated endothelial cells. But for today's talk, I want to turn your attention to a project that we've been building for quite a few years now, ever since I started my lab, which is really focused on the fibrosis resolved liver. And this gets to the idea of cell memory and tissue memory. So this work is led by Alvaro Gonzalez, who is a postdoc in my lab, very first postdoc, so he's pretty brave. And the question that he wanted to ask is the liver, once the fibrosis is resolved and the inflammation resolves, has it really truly returned to baseline? In other words, is it possible that the fibrosis resolved liver actually carry a memory of the previous injury? And what would be consequence of that memory? So to study this fibrosis memory, we first had to make a mouse model that would essentially model what we see in the patients that undergo fibrosis regression. So here we use this chemical toxin called carbon tetrachloride. We gave the mice a few doses of carbon tetrachloride over a few weeks. And then we just terminate the injury and let the fibrosis resolve. So as you can see here, the fibrosis readily resolved within 2 weeks after terminating the injury, again, speaking to the quite amazing recovery ability of the liver. And alongside with that, we see that the stellate cells activate. So this is in the bottom panels. They activate as marked by the alpha-SMA staining and rapidly deactivate. So you see that the alpha-SMA staining goes away. And by 2 weeks, we no longer detect any alpha-SMA positive cells. So then we wanted to ask, what is the consequence of having this fibrosis that has previously taken place, but now have completely resolved in these livers? So to ask this question, we essentially just challenged the mice with additional dose of CCL4. So here you have mice that are uninjured and seeing CCL4 for the first time. And then you have mice that undergone fibrosis regression and now are re-exposed to CCL4, so which we call re-injury. And the question here is that, is it possible that regressed liver, the stellate cells that have deactivated, would now have actually a different response compared to the mice that are exposed to CCL4 for the first time? And the answer is yes. So when we looked at alpha-SMA staining as an indication of stellate cell activation, we see that in the re-injured mice, there is a much stronger alpha-SMA signal compared to the initial injury. So then we wanted to understand a little bit more about this re-injury response. Is it just a hyper activation? So is it just the same activation process, but now gone further, or is it maybe a different response? So to address this, we submitted all these samples for single nucleus RNA-seq, and we were able to extract stellate cells from our single nucleus RNA-seq data. We detected roughly 5,000 stellate cells across all 4 different conditions. And first of all, when we compare the stellate cells from the regressed liver to the uninjured liver, we see that like the histology results suggesting that the liver has returned back to normal, we see that the stellate cells at the gene expression level have also returned back to normal. There's very few genes that remain upregulated in the regressed stellate cells. However, if we look at the re-injury compared to the initial injury mice, what we see is that in the stellate cells, the re-injured mice have a very different transcriptomic response compared to the initial injury. So this suggests to us that the stellate cells are actually activating a different response in response to this re-injury. So to get an idea of what this altered response could be with repeat injury or re-injury, we first wanted to annotate the different stellate cells that we identify. So here we identified a few different clusters of stellate cells, some of them that we have not previously seen and have not been previously reported by others. So we see clearly a quiescent subset, and we see a clearly activated subset. However, we see these novel subsets of stellate cells that emerge with injury and re-injury. And these side trajectories of stellate cells, when we looked at the gene expression of these novel subsets of stellate cells that emerge with injury, we see that the most differential expressed genes in these subsets compared to the rest of the stellate cells fall under vascular phenotype. And there's another subset that have an inflamed phenotype. And finally, we have a subset that have epithelial phenotype. So at this point, I think people that have been following the cancer literature start to kind of ring some bells, because this is kind of what the cancer folks have been reporting for the last few years, that there's this increased plasticity of cancer-associated fibroblasts. And specifically, what is very often detected in different kinds of cancers or different kinds of CAFs are vascular subsets of CAFs and inflamed subsets of CAFs. So we think that perhaps for the stellate cells, which are essentially the fibroblasts in the liver, in response to chronic injury, we also end up with these subsets of stellate cells. So then the question is, this tendency to become or to take on these novel phenotypes or becoming more plastic, how does that change in the initial injury versus re-injury? So we just basically subsetted the cells into the ones that came from the injury mice and the ones came from the re-injury mice. And what we see is that with re-injury, this tendency to engage these alternative subsets of stellate cells, these novel subsets of stellate cells, seems to have become heightened. So there's increased tendency to become more plastic with repeated injury. And we try to validate this with some co-stainings. So we had these M-cherry labeled stellate cell, mice with M-cherry labeled stellate cells, which allowed us to co-localize with other markers, and we co-localize with markers that were identified from these novel subsets, markers of epithelial stellate cells, of our inflamed stellate cells, and of our vascular stellate cells. And in all cases, we see a co-localization of the stellate cell marker or traditional stellate cell marker and these markers of these novel subsets. But coming back to the human patients, because CCL4 is an experimental model, it's very convenient to use in the lab, but is this really happening in the human patients? So we will predict then that in the presence of chronic liver disease, the stellate cells in the human patients will also take on these novel identities and become more plastic. So then to answer this, we essentially mine data from other labs that have been published from human patients. So this is a really nice data set from Gribben et al., where they captured hepatic stellate cells from patients with progressive MASH fibrosis using single nucleus RNA-seq. So we took their data set, we isolated the stellate cells from the data set, and we first did unbiased clustering of all the stellate cells in the middle panel. And then what we did is to project the score made from all these novel subsets of stellate cells we identify in mice, and we projected that onto this unbiased clustering of the human stellate cells. And what we see is that the score we derive from mice very nicely characterize the different types of stellate cells that have emerged in the human samples. And we think this is pretty strong evidence that the stellate cells are also becoming more plastic in human patients with repeated injury in these chronic liver disease, in this case in MASH, and that they also take on similar alternative trajectories, taking on vascular phenotype, taking on an inflamed phenotype, and epithelial phenotype with chronic injury. And we also tried to do some co-staining of human patient samples. It's not as clear because we don't have mCherry labeled human patients. But anyway, we found some markers of stellate cells, and we were able to co-localize with markers of these novel subsets of stellate cells, and we think the data is fairly convincing. So as I mentioned briefly with the cancer, we think this phenomenon is not just in liver fibrosis. We think that fibrosis of other tissues could also become more plastic with repeated injury. So we went ahead and tried to test that in a different model, so in the heart fibrosis, working in collaboration with Joneth from Corey Levine's lab at WashU. What they have essentially is a model of inducing fibrosis in the mouse heart, and then they carried out single nucleus RNA-seq from the healthy or the fibrotic heart. And when we took a look at their data, extracted the fibroblasts from their data sets, and then we compare the fibroblast, the expression of the fibroblast from the healthy controls, what we see is that the top oncology terms from the control fibroblasts are canonical fibroblast markers. So these are markers of mesenchymal cells, the stromal, and the parasite. So these are kind of in line with what we think are normal fibroblasts, how they should be. But however, if we perform a similar gene ontology analysis of the fibroblasts that are coming from the fibrotic condition, this time what we see is that the top terms are actually immune-related cells and also some endothelial cell terms. So what we think is happening to the fibroblasts in the fibrotic condition and the fibrotic heart is that they're also becoming more plastic, and they're taking on these novel identities, the similar vascular and inflamed identity as the human stellate cells and the mouse stellate cells. So in the last slide, I just want to show you some work that's currently underway, where we're trying to understand the molecular basis of this increase in plasticity. So as I showed you, we think that going from injury to re-injury, you have increase in plasticity of the stellate cells. But what leads to this increase in plasticity? And as I've shown you before, when we look at the gene expression of the stellate cells in the regressed liver versus the uninjured liver, we didn't see that many gene expression differences. We see some genes that are more repressed in the regressed stellate cells, but we didn't see many genes that were overexpressed in the regressed stellate cells. Again, showing that these stellate cells in the regressed liver have more or less returned back to a quiescent or baseline condition. Then I think it's very natural to assume that maybe there's an additional layer of regulation that could actually store this previous injury memory and translate that into a future maladaptive response in the form of increased plasticity. So to test this, we performed single nucleus RNA attack-seq, and which is a way of assessing the global chromatin accessibility in the stellate cells. And in contrast to what we've seen before with the gene expression data, we see that now with the attack data, we see many more peaks. So these are regions in the chromatin that have demonstrated different accessibility, indicative of some epigenetic change. We see many more peaks that are differentially accessible in the regressed stellate cells compared to the uninjured stellate cells. So the further next step for us would be to try to link the two observations. So one is an increase in epigenetic changes in the regressed liver, and another one is increase in stellate plasticity. And to do this, we did some data mining. So we took the peaks that are upregulated in the regressed mice, and we mined it for motifs or transcription factors, which we think would be the potential mediators of this epigenetic memory, and translate that into a gene expression difference in terms of increased plasticity. So when we did this motif mining, we identified a few different transcription factors that were already established and known to be involved in fibrosis in the liver. However, we came up with a new one that has never been, to our knowledge, described for liver biology at all, which is TBX20. And in fact, we see that TBX20, depends on which algorithm you use, is present in 10 to 20% of all the motifs that have been extracted from the differential accessibility data. So what we're currently working on now is to, again, turn back to our mouse models and inducibly delete TBX20 in the various conditions. What we're hoping to show is that TBX20 could read this change in the epigenetic landscape in the regressed mice and translate that into a change in the plasticity of the stellate cells. And that is something that we're currently working on. So just to give you a summary of what we found. So we found that even when the fibrosis resolves, the stellate cells could deactivate. However, this deactivation is incomplete at the level of the epigenetics, and that there are these regions in chromatin that have changed in terms of their epigenetic landscape that does not translate into gene expression right away. However, these cells are primed for an activation. And in the presence of re-injury, for example, when you have a Thanksgiving dinner or when you have the wine and cheese later on, you trigger these deactivated stellate cells, and that leads to hyperactivation of these cells. And that over time of chronic injury and chronic disease, this epigenetic memory would most likely become more and more entrenched, and that sets up for this altered response in the repeat or in the chronic liver disease that we see with the stellate cells, where they're increasingly prone to produce more and more extracellular matrix that make up the fibrosis. And furthermore, we think that there's a specific mediator, there's specific transcription factors like TBX20 that can read this change in the epigenetics and translate that into a change in the behavior of the stellate cells in response to re-injury, and leading to overall an increase in plasticity of these cells over time of injury. And we think that this is perhaps a conserved mechanism across different tissues. This idea of having fibrogenic memory that could be stored in the fibroblast on the level of the epigenetic changes. So with that, I'd just like to thank everybody in my lab, but mostly Varo for taking on this very challenging project, and also our collaborators for this project and current funding sources. Thank you very much.
Thank you, Dr. Wong, for that wonderful talk. Are there any questions from the audience?
Hi, Sammy. Cool talk.
Thank you.
So your epigenetic data definitely argued for a cell autonomous effect, but I was thinking about the non-cell autonomous effect, since we know that HSCs and T cells interact a lot.
Yeah.
I'm wondering if you have any thoughts on that?
Yeah, really good catch. So one of the assumptions we're making is that there's something inherently changed in the fibroblast, and because they can deactivate and stay around for a long time, they could carry this epigenetic memory. But what we haven't ruled out is some kind of immune memory that could also play a role, like maybe some immune cells are long-lasting and retain in the liver. All I can say is that histologically, we don't see many immune cells anymore. When the fibrosis resolves, the liver looks almost pristine, at least at the level of histology.
Now we have one from Zoom. So great talk and beautiful science. Is there any spatial heterogeneity you see with the hepatic stellate cell plasticity?
Yeah, so that's a great question. So this kind of gets at a different project we have in the lab. So basically what labs like Robert Schwabe's lab have found recently is that depends on where the stellate cell is, it could be beneficial for the liver or detrimental. So so one of the future directions is actually try to map these subsets of cells to different locations in the liver and link more, the spatial aspect of it. So for sure, that's on the to-do list.
Have you tried to vary the time course to the re-injury and see if there's actually recovery if you wait long enough? And if there's repeat injury, does it
Yeah.
affect the time course?
Yeah, so one of the things we don't know is how long does the epigenetic memory last? So we see the gene expression has gone back to baseline. We think that eventually the epigenetic changes probably will also revert back or get lost, but I think the time scale is longer. So even when the gene expression is returned quickly, the epigenetic changes seems to be more long lasting, but we haven't tested how long lasting that is because I think in the real case or in the real world scenario, you essentially have a continuous accumulation of the epigenetic changes, unfortunately. So you never really recover completely, if that makes sense.
Okay, our final speaker in today's session is Dr. Robert Langer, who needs no introduction, but I will do my best. He is one of 9 institute professors at MIT, which is MIT's highest honor that it confers to its faculty. His articles have been cited nearly 500,000 times, and he is the most cited engineer in history. He has, along with that citation track record, a long list, more than 220 awards that he's received. He holds 44 honorary doctorates, and he is one of 3 people that are currently living who have received the United States National Medals of Science, Technology, and Innovation. And he is also an elected member of the National Academy of Medicine, National Academies, plural, of Medicine, Engineering, Sciences, and Inventors. He has also had a long track record of industry collaboration. His patents have been licensed or sub-licensed to over 400 companies, and he is a co-founder of many of those companies, including Moderna. So without further ado, Dr. Langer.
Thank you very much. You know, when I was thinking, I don't know, it was maybe many years ago, you invited me to speak at this, and I remember that fondly, and I feel very happy that Sangeeta invited me to speak today. You know, it's certainly remarkable what you've done, and you know, anyhow, my talk probably will be a little different. I thought I'd tell you a little bit about. I mean, I will mention the liver a few times, but it's not really been the main thing that I've focused on. But I thought I would tell you a little bit about how I, as an engineer, got into this area of medical science. So I got my degree from MIT in chemical engineering in 1974. Those days, you know, which was over 50 years ago, chemical engineers pretty much went into the oil industry. I thought I'd do that too, since everybody else all my friends were doing that. But you know, when I went, I actually got quite a few job offers because anybody could. You didn't have to be very good if you left MIT and you had a background in that. So I got quite a few offers, but I was very disappointed when I'd go to these interviews. You know, they'd be telling you how if you just could increase the yield of certain petrochemicals by tiny percentages, how wonderful that would be. And I thought to myself, I didn't really want to spend my life doing that. And it made me think what I really did want to do. And when I was supposed to be doing my PhD thesis, one of the things I got very involved in, this was now in the early 1970s, was helping start a alternative high school for low-income students. You probably all don't realize, being in Cambridge today, that Cambridge in 1971 had the highest dropout rate for high schools of any city its size in the country. It's over 35%. So I got very involved in creating new chemistry and math curriculum. And one day I actually saw an ad in Chemical Engineering News that City College of New York had an opening for somebody to be an assistant professor to develop new chemistry curriculum. I wrote them a letter applying for a job, but they didn't write me back. But I really liked that idea, so I started thinking about other places, and I actually found about 40 colleges that had positioned something like that, and I wrote them, and none of them wrote me back. So that wasn't going so well. So I started to think, well, what other way could I use my chemical engineering background to help people? So I thought about medicine. I wrote to a lot of hospitals and medical schools. They didn't write me back either. And then one day I was walking into the lab I was at, is building 12 here, and one of the older scientists said to me, he said, Bob, he said, there's a surgeon in Boston named Judah Folkman. And he said, sometimes he hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. But Dr. Folkman had this idea, and he was kind enough to offer me a job. It didn't pay like the oil companies, but I was very excited about it. And he had this idea that if you could stop blood vessels, that might be a whole new way to someday think about treating cancer. I thought it was a fascinating idea, especially since I didn't know much about biology. It turns out he was widely criticized for that idea. By the way, the other thing that I thought was, he was, to me, a very inspirational figure. I still remember, he'd give me these books, the one about Howard Florey and penicillin, and he did give me a book on the liver, and we were so fascinated about it because, it was the one you could, take so much of it out of the body and it would just grow right back. He said, someday people will figure out why. I really loved working there. I was the only engineer there for a number of years. And the problem I was working on was to prove he was right, that this really could work and that blood vessel inhibitors did exist, even though journals like Science and others said they could not. So one of the keys, maybe the key to solving this problem was there was, at that time, hardly any way to study blood vessels. The biggest problem is there's always background blood vessels. So a couple of places where they're not. One is the chick chorioontoic membrane, where there are very few. The other is a cornea. And so our idea was we put little tumors in, like a V2 carcinoma, and that will, as I'll show you, bring in blood vessels. But could we create a tiny little material, like a delivery system, like a nanoparticle or a microparticle, that could deliver the different molecules we were isolating? And at that time, and all the molecules we were isolating were large molecules, like proteins, peptides, polyproteoglycans, nucleic acids, things like that. So at that time, the idea of drug delivery, this is 1974, advanced drug delivery systems was pretty much unheard of. There was only one company in the world then, a company called Alza, that was doing anything on drug delivery systems. And they hadn't commercialized anything yet, but they were making advances. And in fact, they would later on come out with all these transdermal delivery systems that have been very successful today. But Dr. Folkman went to see them and said, well, could they help us? But they all said, well, you really can't deliver large molecules. They're too big. They can't get out, or if they got out, they'd get out right away. So that was kind of discouraging. But if we were going to solve the blood vessel problem, we had to solve this problem. So I spent several years working in the lab, experimenting with different materials, different designs, and so forth. And I found actually over 200 different ways to get this to not work. But eventually, we were able to make these little particles. Here's a picture of one and one cut in half. And we published in Nature that in 1976 that you could use this approach to deliver any large molecule. It's also the earliest paper ever showing that it was possible to deliver nucleic acids from any tiny particle. So I want to go back to 3 months before this paper came out. I got asked to give a talk, a 20-minute talk at a big symposium in Midland, Michigan. I was 27 years old, and I was a terrible speaker. I practiced this talk for weeks. Finally got up and I gave the talk. And, there were all these well-known chemists and chemical engineers. And I thought when I was done with the talk, I actually did all right. I didn't forget that much of what I was going to say, and I didn't stammer too much. So I thought all these older scientists, being nice people, would want to encourage me, this young guy. Shows how much I knew about science. But at any rate, so when I got off the podium, I stepped down, and a whole bunch of them came up to me, and they said, we don't believe anything you just said. Again, they said the molecules were too big, and they also said the solvents we were using, like methylene chloride or ethanol, would destroy whatever we put in. And things actually went downhill from there. I tried to get research grants, and my first 9 were rejected. Actually pretty badly. They would go to what are called pathology B study sections, and they said, like, if you're an engineer, you don't know anything about biology or cancer. So I did very badly on that. And then, I loved being at Children's Hospital. I really changed my life. But a lot of my friends, talked to me and they said, Bob, you probably shouldn't be a postdoc your whole life. So I did. I started applying to faculty positions, and I a chemical engineer, so I applied to chemical engineering departments. But no department in the world would hire me. I got some interviews, but they would come back and saying, these bio ideas that you're talking about, chemical engineers doesn't make sense, like drug delivery or blood vessel growth or things like that. So I didn't get any jobs. Finally, Dr. Folkman introduced me to Nevin Scrimshaw. Nevin Scrimshaw in the 1970s was one of the most famous nutritionists in the world. And he was the head of the nutrition department at MIT. He also was what I call a benevolent dictator, a kind of department head, which what I mean by that is, for me, I thought good. He liked me, so he offered me a job, but he didn't ask anybody else in the department what they thought, which would have been okay, except for one thing, which was the year after I joined the department, he left. So a lot of the senior faculty decided that I should leave too. And just to prove to you that, a few years ago, maybe 12 years ago, I was getting this award, and Mike Marletta, who was a colleague of mine at the time, Mike later would become president of Scripps, head of Berkeley's chemistry. This is just an excerpt, but he was talking about what it was like to be with me in 1979. And he said one evening he went to faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said a senior scientist said, quizzing us while smoking a cigar. He said when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. So I had a pretty rocky start, but I kept at it, and as I mentioned, since we had done the delivery work, now we could try to address the angiogenesis problem. This is the assay that I mentioned. I'm going to show you kind of a gruesome slide, but there's no other way to show blood vessels. So I'm going to show you some corneas. This is from a paper we wrote in Science just about 50 years ago. And there are, if you look on the left-hand side, you can see that the blood vessels grow as like a sheet. But on the right, and we probably did over 1,000 of these. I worked with several surgeons, Mike Klein and Ken Falterman. But on the right, and we had lots of different fractions. I think over 100 of them didn't work, but one did. And we repeated this over 20 times. And you always would get this, and the vessels were lower, they were sparser, and they would actually avoid the tumor. So this did show for the first time that blood vessel inhibitors could exist, and it would provide a pathway to this day, actually, for future inhibitors to be tested. And of course, what's happened, and it took a long time. This was a 1976 paper. It took 28 years from this paper, and really great work from Genentech and other places. And this may be hard to see, but what happened is starting in 2004, Avastin gets approved. Avastin is probably the #2 best-selling biotech drug in history. Even last year, it was the 4th biggest selling drug in the world. But you see many different ones getting approved. Journal Nature estimates that 500 million people will be treated with these. And the thinking is that, and what's happened is it's not only being widely used for cancer treatments, but it's really the only pharmacologic treatment for diseases of blindness in the back of the eye, drugs like Eylea and Lucentis. Well, I want to go back, so that takes you up even to today, with I think 4 of these getting approved in the past year. But I want to go back to the 1970s again. And one of the things that Dr. Folkman said to me one day was, we should file for a patent. So people that know me, they probably think, well, that would be obvious, but it wasn't. I had 0 patents then. And so did Children's Hospital. They didn't have any. So we applied for one for the first time. And 5 years in a row, it got rejected. So I remember Al Brazaghini, who was the head of tech transfer at Children's Hospital. He came to see me one day. He said, Bob, you have to stop. You're just wasting so much money for the hospital. They're never going to allow the patent. He said, you should just quit. But I don't like to quit. So I started to think, how could we convince the patent examiner to allow this patent legally, of course? And science wasn't really working. But as I told you in the beginning, when I first started doing this, everyone said it was impossible. It could never work. I wondered, maybe somebody wrote that down. So in 1982, after getting 5 years of rejections, I did what's called a science citation search. And I found a number of articles, but I just want to show you a quote from one of them. And these were at that time very famous engineers and chemists. And they were just writing a review in 1979. I had no idea that they did this, but it was very helpful. They wrote describing this field. Generally, the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules could be released by such a technique because of extremely small permeation rates. However, Folkman and myself have reported some surprising results that clearly demonstrate the opposite. So I knew enough about patents to know that that word surprising was good. So I showed it to Dr. Brazaghini. He flew to Washington, showed it to the examiner, and the examiner said he had no idea. He said, tell you what, I will allow this patent if Dr. Langer can get written affidavits from each of these 5 people that they really wrote that. So I was assistant professor. I wrote them, and they were all nice enough to write me back. They really wrote it. And we got this very broad patent. And you know, I've been working on this now for about 10 years, and I always had this dream that something I did might someday be useful. And that really wasn't happening. I mean, we were writing papers and people, you know, would quote them and stuff like that. But I really wanted, you know, products, something that would happen. So finally, in 1984, I started getting, 10 years after I had started, I'd got some calls from some multi-billion dollar companies, one in animal health, one in human health. And they said, gee, we've read your work. Would you consult for us? And, you know, what we'd like to do is give you a big grant, give you a consulting fee, and we want to develop these into products. And I thought this was terrific. It was great getting the grant money and everything else. And I had a Ford Pinto at the time, wasn't a very good car, so I finally could get a new car. At any rate, so what happened was those 2 companies, each of them, you know, worked on it for a year or 2, and then they just gave up, which really made me sad, in spite of the car and grant. But at any rate, a couple of years later, Alex Klebanoff, one of my good friends, he said, Bob, we should start our own company. So we did. We started this little company with 4 of our students. You know, and I've kept doing that the rest of my life. What I found is if you're not your own champion, nobody else will be. For most everything, every so often, you know, people do stuff. So these are some of the things that came from it, and there are all kinds of different products. You know, and I've been, you know, on different boards, founding companies. From the liver standpoint, of course, they're one of the sponsors of this. Alnylam, I was on the founding scientific advisory board. Akina Kink, one of my students, was the number 1 or 2 employee, and one of the exciting things that they did is develop little nanoparticles that could deliver large molecules, in this case siRNA, and direct it to the liver by putting GalNAc on the surface and targeting it to the acylglycoproteins on the hepatocytes. And that was a real big deal, and it got approved in 2018. That's on Patro. And now Alnylam has, you know, been an incredibly successful company. Another example was Moderna, or is Moderna, but there are many, many others, and they're not just limited to medicine. Yanni Zohar, who came to our lab, started applying it to aquaculture, and partly because of him, that's now a $250 billion industry. So, so that's kind of how I got started. But there was one other area that I also started getting involved in, and that was new materials and new tissues and organs. And one of the good things about working in the hospital is that it gave me a window into seeing how materials found their way into medicine. And I thought, again, being very naive, that it was probably older chemists or engineers or people like that. But really, almost always, if you look at the 20th century, the way materials found their way into medicine were medical doctors going to their house to find some object that kind of resembles the organ or tissue they want to fix. So if you look at the artificial heart, what happened is some of the surgeons, they said, well, if we want to develop a heart, we need something with a good flex life. So they asked what object has that, picked a lady's girdle. Problem with that, and it made progress, but the problem with that is sometimes blood hits the surface, it does hit the surface of the artificial heart, forms a clot, and then goes to the brain, and the patient gets a stroke and they die. breast implants. One of those was a mattress stuffing because it's squishy. Anyhow, this was really, if you look at almost the entire 20th century, what people did. I started thinking about this in different ways. First, being a chemical engineer, maybe you could ask the question, what kinds of materials would you really want? And then design them from first principles. That's not part of the talk today, but the other thing we thought about working with Jay Vakanti is, well, maybe we could make, use it to combine with cells and make new tissues or organs. And actually what motivated that was was the head of the liver transplant program at Boston Children's Hospital when he first came to see me in the early 1980s. And he saw little babies like this one who was dying of liver failure. And the only way a child like that would live is somebody else would die and Jay would get up in the middle of the night and do a transplant. So we started talking about this and ultimately came up with this paradigm that you could combine different cells with materials in 3 dimensions and do something like this and make any kind of tissue. So we'd make them into different fibers with cells. And I thought I would just give a couple of examples of what you could do, just at a very high level. Well, one that happened, which we licensed to a company, a couple of companies, was burns. So here's a little baby who's badly burned. You can take the product, which is neonatal skin fibroblasts, on a scaffold. You can put it on the child at the time of injury. But if you come back 3 weeks later, he looks like this. And 6 months later, like this. And then of course, the other thing that this kind of work leads to, and Sangeeta has done, really pioneering work on this, and I'll mention some of the people in our lab too. Gordana Vanyak and Milika Retzik, they wanted to make a heart on a chip. And this is just a high-level example where you put cardiac muscle cells on this, and you get this cardiac-specific ultrastructure. And this is a video of 6 million heart cells beating. We're not shaking it or anything, but it's really beating. And you can even take an EKG of it. And actually, Milika and Gordana even started a company on it that worked on it with quite a few other companies for drug testing. So on the very last slide, I thought I would just show you where we are today, both of the people that have worked in our lab and who have left our lab. But there are many different tissues and organs that they've done. We had grants from the Army to make new ears for soldiers returning from the wars. We worked with different people to even make spinal cords that got into clinical trials. On organs on a chip, we've met Gio Traverso, who we do a lot of work with, who's now a professor at MIT, actually made a GI tract on a chip, which, again, one of the things you can use is really figure out how to increase absorption by doing high throughput screening. And then you see the heart on the chip. Most recently, which I've been very excited about, Li Wei Sai, who heads the PIC Hour, contacted me about 5 years ago, and she said, well, can we write a grant to make a brain on a chip? So we were able to actually get that grant. And I had a terrific postdoc from Stanford, Alice Stanton, and she made that. It just came out in PNAS with 6 different brain cell types and a blood-brain barrier. But again, things like that might be useful for testing. And actually, Mass General just started a new neurology institute, and she's the first faculty member they hired. Anyhow, going back to other tissues and organs, we've worked on hearing. I mentioned liver. Another person we've collaborated with is Steve Zeitel. Steve is actually the voice surgeon to the stars. And so you see a picture, that's me with Julie Andrews, if you look closely, who's one of his patients. And if you ever, I don't know if anybody runs, but if you run, you bang, there's a lot of beating of your knee and feet and stuff. Well, if you look at videos of Julie Andrews singing, the vocal cords, enormous, banging. So you get a lot of scar tissue. And so she really is no longer able to sing. I mean, so anyhow, we're working on new vocal cords. I mentioned skin, and with Dan Anderson and the Juvenile Diabetes Foundation, we're working on pancreases. So I want to thank Sangeeta for inviting me. I want to congratulate Wynn. I thank you for staying so late and listening. And thanks so much.
Thank you, Dr. Langer, for that truly inspirational talk. Does anyone have any questions or anything from the audience?
Hi, thanks, Bob. So inspiring always to hear you. I was wondering if you can touch on the concepts of function in the context of tissue fabrication or creating organs.
Function.
Function.
Yeah, well, that's kind of important. But I mean, I think, well, what you can see, I mean, that with some of these different things, I mean, in a way, like say, pick the heart on a chip. I mean, you can actually see that it beats, so you can see function. What I didn't mention, and I probably should have, though, this is a really good example on that last slide, as I think I bypassed, didn't go over blood vessels, which is really exciting. This was work that Laura Nicholson did when she was in our lab, and we published this in Science in 1999. And I'm going to put that back on, and I'll show an example. But anyhow, what that's led to is she became a professor at Duke and then at Yale, and then started a company called UMass. And they actually got FDA approval of really the first synthetic small diameter blood vessel. And you know, that's actually used now to treat Ukraine soldiers. There was a major publication on that was just out a few weeks ago. So I think certainly in the case of heart and blood vessels, there's some function. You have to sort of look at what what kind of markers would tell you function. We did some things on hearing, and there again, we could see some function, but it depends on what you're looking at.
What about the liver?
On the liver, well, liver, unfortunately, we haven't done as well, but fortunately, people like Sangeeta and others have done a good job. But I would say we really have never, I think, succeeded enough on that. There were a variety of people that trained with us, where they try to make extracorporeal livers, but I don't know that any of them ever, really made it. They were in clinical trials, but they never really led to products. I'd like to think that they had some function, but I don't know if they had enough function, and so that's it's unclear. But liver, Jay and I started working on it, and he's still probably working on it in certain ways. That was what got him started.
Bob, you actually spoke twice before in this program. The first time was in 1998, and the second time in 2005. So
thank you. Okay, your memory is better than mine.
But so I'm really curious. To what extent has serendipity played a part in your career? And do you have any suggestions for young people who are confronting all the hazards and problems of today, not just the political ones, but scientific ones?
Yeah, well,
how do you sustain motivation when there's so much negativity?
Yeah, well, it's a great question. I think serendipity certainly played a role in what I did. I felt lucky, though. I would say, whether it's me or anybody, I used to play golf, and I remember when I was much younger, and I hit a bad shot, and I would always tell my father that I have bad luck, and that's why I hit the bad shot. And then my father would always tell me, well, Bob, you make your own luck. And so I think there's some truth in both of those things. I think, for me, certainly working with Dr. Folkman at Boston Children's Hospital, that was serendipity. But, I took I didn't do what everybody else did, just because I didn't care. It just didn't matter to me at that time about making money or anything else. So I don't know. So on that, and to your second question, I just think, there's sometimes things are harder than others. I've often said that the people that are most successful are the ones that know how to deal with failure well, because success, that's easy to deal with. And so, you know, you have to sort of put your head down and when things are tough and you keep trying. You know, so, but, and there's always, everybody has ups and downs. I'd like to hope that, you know, things get better, but I don't know financially whether they will or not, or certainly right away. But still, I think still I see people still doing great work, and I believe anybody can. I'm an optimist.
Before we get to the awards, I just want to thank everyone for contributing to this fantastic symposium. Most of all of you attendees for being here, staying here, and with your questions, making this really a truly interactive event. I want to thank Win and for really not just inspiring us for all 34 years and before, but really for teaching us today and enlightening us about again about the liver and bile acids. And for all of our speakers today, we went from looking very deep into the cell, into the ER, to looking at organs and organisms and populations. We thought about how we can take wisdoms from our patients and bring it back to patients in Bangladesh and in East Boston. We thought about how we develop organs and how organs and organisms age. And I think, like Sangeeta, I was a medical student when I went to the second Arias Symposium and inspired me about the liver. And now I'm obviously much, much older. And this symposium still inspired me, and it's all of you who inspired that. I want to thank my colleague and friend Sangeeta and Heather for putting this day together. I wanted to thank Moore, who has been helping at the front desk to make sure our registrations and program really went over so well. Want to thank our host here, the Broad Institute, for bringing us together at really the epicenter of science and technology, innovation, and translation. And I wanted to now announce the poster award winners. We have 2 categories. One, our judges, and I want to thank all of them who went around during lunch break, talked to our trainees, and helped us find the winners. And then the fan favorites, which is all of us, and that's fantastic too. I do think it's really the trainees, and we just heard that in the last question from Win to Bob. It's the trainees that give us our future, right? At some point, we'll all age, Vadim, despite all your efforts, and we'll become patients. And so then we need the next generation that is in this room to both help us and our livers, but also keep pioneering and reinventing the field. So the third prize for the judges award is Florian Hügeler from the Tibet lab all the way from Zurich. The second prize is Chao Dong from Sammy Wang's lab at Montpellier. And the first prize is Vardman Kumar from Sangeeta's lab across the street. For the fan favorites, we have a tie, and I promise you I didn't vote. It is for Marcel de Carvalho Ribeiro from John G. Zabo's lab at the BIDMC, and Lily Mersheimer from our lab here at the Big. And with that, I hand over, and I wanted to thank our host, sponsor, and initiator of not just this symposium, but really an organization that is with us not only when we do research in the lab, but also helps our patients and inspires, educates them. And that's the American Liver Foundation and Lauren Steele. Thank you.
So what another exciting program. I've had the honor of attending so many of these since COVID, and I've just been so grateful. And of course, we are so proud that this program exists because of the amazing Dr. Irwin Arias, who had this vision all these years ago. And we are so grateful, and Wynn, you've been a mentor for me since the first day I became CEO of the American Liver Foundation, and I will never forget you and all the learning that I have had the benefit of receiving from you. I just wanted to give a couple of points about what's happening at the American Liver Foundation. I have talked to so many of you. You know, we reinvented ourselves after COVID, and very exciting. We have funded $2.5 million in research in just over 2 years, funding many of you in this room because we feel support of basic science research is so important. So how about a round of applause for all of that support for all of you, and it's going to continue to move forward. You can count on the American Liver Foundation. I also wanted to share that we launched a patient registry a couple of years ago, and we have over 1,000 people who have participated, and we see great growth in that. We hope that will be a resource for all of you. And also, most recently, October 6th, we launched a non-directed living liver donor transplant network, a registry for people who might want to give part of their liver and to someone maybe they don't know. And how can they do this? We made it work for them. We have 35 transplant centers on the list with us, available to those who want to make a donation and save a life. So we're really proud at the American Liver Foundation that we meet needs, we fill gaps. I want to acknowledge Dr. Len Jordan on our team, who leads our research effort. I know many of you are in constant contact with her, and she's been with us all day, but from afar, so thank you, Len. A few other thank yous. Heather, the unbelievable Heather. None of this would happen without Heather. We're so grateful. Another round of applause. Sangeeta and Wolfram, oh my gosh, you really are the heart and soul of this program, and we're so appreciative of what you do here. We want to thank our presenting sponsor, Alnylam. They have been very supportive, as well as other sponsors, including Wynn Arias and Luba and Wynn's sister Joan Gordon, who's been supportive of this program for many, many years. Also, a big shout out to an AV, you know, audio visuals, everything, right? You have a program like today and you're like praying every moment that you're not going to have any challenges. And we're very appreciative of Owen Pappas Bigelbach from MIT, who has been our AV person today. So thank you, thank you. No mishaps, no problems. That's a good day. And of course, all of our speakers were exceptional. And you were all exceptional with great questions. And I can't wait to get to know more of you. And thanks to Heather, we have a tremendous cocktail party next. We hope that you'll all hang out and enjoy, and we can talk about our day. So thank you from the bottom of our heart for participating in this program. Take care.